there's a fish that can see through its own head. Not metaphorically, literally. Right now, 2,000 feet beneath the Pacific, a creature hovers in absolute darkness with eyes embedded in a transparent skull. Tubular eyes, green, glowing, rotating inside a dome of clear tissue like organic telescopes scanning for prey in a world where sight shouldn't be possible. This isn't science fiction, it's the barreleye fish, and it's just the beginning. Because the ocean doesn't follow the rules we wrote for life. Down there in the crushing pressure and eternal night, evolution took a different path. A stranger path. Creatures exist that shouldn't be able to, with transparent bodies, bioluminescent organs, eyes the size of dinner plates on bodies 6 inches long, blood that's blue instead of red, metabolism so slow they can survive for months without food. They don't just look alien, they are alien. Their chemistry is different, their physics is different. The pressure that would crush a submarine is the pressure they need to stay alive. The darkness we fear is the light they avoid. The oxygen-depleted water that would suffocate us in seconds is the air they breathe. These aren't Earth creatures that adapted to the deep sea. These are deep-sea creatures that happen to share our planet. Tonight, we're diving into a world that breaks every biological rule we thought was absolute. Where evolution didn't just adapt, it invented. Where life found ways to exist that we're only beginning to understand. This is Omni. Stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a moment to 500 feet below the surface. No light, no warmth, pressure so intense it would collapse your lungs in seconds. And in this darkness, a creature hangs motionless, waiting. It's the barreleye fish, Macropinna microstoma, and everything about it is wrong. Its head is a clear dome, not skin, not scales. A transparent shield filled with fluid, and inside that shield, 2 bright green cylinders point upward. Those are its eyes, not where you'd expect eyes to be, those dark spots on its face and nostrils. No, this fish's actual eyes are behind its face, embedded in its skull, free to rotate like a submarine's periscope. For 70 years after scientists first netted a barreleye in 1939, we thought we understood it. We thought those tubular eyes were fixed, locked in an upward gaze, giving the fish only tunnel vision of whatever drifted overhead. We were wrong. In 2009, researchers at the Monterey Bay Aquarium Research Institute did something remarkable. They brought a living barreleye to the surface and kept it alive long enough to watch. And what they saw changed everything. The eyes rotated. When the fish tilted its body from horizontal to vertical, the eyes turned with surgical precision, shifting from looking up to looking forward. According to marine biologist Bruce Robertson, who led the study, this transparent dome isn't just protection, it's a lens. It allows the fish to scan the water above for the faint silhouettes of prey, then rotate its eyes forward to actually catch and eat what it finds. But here's what makes this truly alien. The barreleye doesn't hunt in the open water. It hovers beneath massive jellyfish-like creatures called siphonophores, colonial organisms that trail stinging tentacles up to 130 feet long, like living drift nets. The barreleye carefully maneuvers through these forests of poison, stealing food that other animals have been caught and paralyzed. Its transparent shield protects those delicate rotating eyes from the stinging cells that would blind anything else. Think about what that means. This fish evolved a see-through head, rotating telescope eyes with bright green filters to distinguish sunlight from bioluminescence, and the spatial awareness to navigate through a maze of invisible, venomous tentacles in pitch-black water, all to steal someone else's lunch. That's not adaptation. That's invention. And the barreleye isn't alone. The deep ocean is filled with creatures whose very existence challenges what we thought life required. 3,000 feet down, in waters so depleted of oxygen that most fish would suffocate, the vampire squid thrives. Vampirateuthis infernalis, literally vampire squid from hell, but it doesn't suck blood. It doesn't hunt. It drifts through oxygen minimum zones where the concentration of dissolved oxygen drops below 20 micromolar, less than 2% of what surface waters contain. To put that in perspective, you'd pass out in seconds if your blood oxygen dropped that low. Most ocean predators can't even enter these zones, but the vampire squid calls it home. How? Its blood is blue. Not red. It uses hemocyanin instead of hemoglobin to transport oxygen, and that copper-based molecule binds to oxygen with such efficiency that the vampire squid can extract every last molecule from water that would leave other creatures gasping. Its gills have evolved massive surface area. Its metabolism runs slower than any other cephalopods. It moves like it's conserving energy for a marathon that never ends. And when threatened, because even in the oxygen minimum zone predators exist, the vampire squid doesn't use ink. It has no ink sac. Instead, it expels a cloud of bioluminescent mucus, a sticky glowing mist filled with thousands of blue lights that pulse and shimmer in the darkness. According to research from the Monterey Bay Aquarium Research Institute in 2012, this luminous cloud can last nearly 10 minutes, and it serves two purposes. It blinds the predator with sudden light in a world of total darkness, and it sticks to the predator's body, making them glow, turning the hunter into a beacon for anything larger lurking nearby. The vampire squid has also evolved another defense. When disturbed, it inverts itself, turning inside out, wrapping its webbed arms up and over its body to expose rows of fleshy spines called cirri. It transforms from a soft, vulnerable creature into something that looks like a spiky alien pumpkin. And if that's not enough, it can turn off every bioluminescent organ on its body at will, becoming completely invisible in the dark. But here's what's truly remarkable. Unlike most cephalopods that live for 1 to 3 years and reproduce once before dying, vampire squids may live up to a decade. Female vampire squids can reproduce 20 times or more throughout their lifetime. They've evolved not just to survive in an environment that shouldn't support complex life. They've evolved to thrive there, slowly, patiently, across lifespans that dwarf their shallow water cousins. This is life playing by different rules, not bending the rules, rewriting them. And it's not just a few bizarre outliers, it's systemic. It's everywhere you look below a certain depth. According to a 2017 study published in Scientific Reports, researchers analysed over 350,000 observations from remotely operated vehicles, surveying the California coast from the surface down to 3,900 metres. They found that 76% of all observed organisms had bioluminescence capability, not 76% of species, 76% of individual creatures they encountered. In some parts of the ocean, particularly below 1,500 feet, that number climbs to nearly 90%. 9 out of 10 creatures you'd meet can create their own light through chemical reactions. It's not a rare adaptation, it's the dominant trait. Bioluminescence has evolved independently at least 40 separate times in marine organisms, 40 different. Lineages of life looked at the same problem: how do you see, communicate, hunt or hide in absolute darkness, and arrived at the same solution: make your own light. But they didn't all do it the same way. Some use bacterial symbionts. They carry colonies of glowing bacteria in specialised light organs. Others produce the light themselves through luciferin-luciferous reactions. Some glow blue-green because those wavelengths travel farthest through water. Others glow red, a colour most deep-sea creatures can't even see, giving them a private channel of communication and a hunting advantage like infrared night vision. The dragonfish, for instance, has evolved red bioluminescence and the rare ability to detect it. According to research published in Science in 2005, these fish essentially have built-in night vision goggles, allowing them to see prey that remain invisible to everyone else. This isn't just evolution fine-tuning traits. This is biochemistry inventing solutions to problems that don't exist on land. This is life under pressure, literal crushing, bone-shattering pressure, discovering that the laws of biology we learned in surface ecosystems don't apply down here. At 10,000 feet, the pressure reaches 300 atmospheres. That's 4,400 pounds per square inch. Imagine the entire NFL and NBA standing on your chest. That kind of force should collapse cells, denature proteins, crush membranes. And yet, life found a way. Deep-sea creatures have evolved proteins that maintain their shape under pressure. They've developed cell membranes with unique lipid compositions that stay fluid when they should be rigid. Some fish, like the Hadal snailfish found in the Mariana Trench at depths exceeding 26,000 feet, have evolved gelatinous, almost boneless bodies. Their flesh is so soft, so water-saturated, that when brought to the surface they lose all structure. According to a 2023 study published in eLife, these snailfish have lost genes for pigmentation, skeletal calcification, and even some visual wavelengths. They've shed every adaptation that doesn't serve them in the deepest trenches on Earth. What remains is streamlined for one thing: surviving where nothing else can. But it gets stranger. Some deep-sea creatures don't just survive the pressure, they die without it. Researchers have tried bringing deep-sea bacteria to the surface. Some species, collected from more than 6 miles down in the Mariana Trench, where pressure exceeds 1,000 atmospheres, simply won't grow at normal surface pressure. They've evolved so completely for the deep that our world is as hostile to them as theirs is to us. Think about that. There are organisms on this planet for whom our environment, the surface, with its breathable air and gentle pressure and sunlight, is lethal. We've created 2 Earths on one planet, and the one below the waves is larger. The ocean covers 71% of Earth's surface. The average depth is 12,100 feet. By volume, the deep sea, everything below 3,300 feet, is the largest living space on the planet. It's not an edge case, it's not a marginal habitat, it's the dominant ecosystem, and almost everything living there has had to become something we wouldn't recognize as possible. There are fish with antifreeze proteins in their blood to survive near freezing temperatures. There are worms that live in toxic sulfide plumes from underwater volcanoes, farming bacteria that convert poison into food. There are jellies made of 99% water that drift through the water column, with such minimal body structure they're barely more than organized liquid. There's the giant isopod, a creature that looks like a pill bug the size of a football, with a metabolism so slow it can go 5 years without eating. There's the gulper eel with a mouth larger than its entire body, hinged jaws that can unhinge to swallow prey twice its size. There's the anglerfish, where the male, a tiny parasite, fuses permanently to the female's body, dissolving until only his reproductive organs remain, a living sperm bank grafted to her flesh. These aren't just strange animals, these are fundamentally different solutions to the problem of existence. On land, life evolved within a narrow band of conditions: temperature, pressure, oxygen, light. They all stay relatively stable. Evolution on land is about competing for resources within those constraints. But in the ocean, especially the deep ocean, those constraints don't exist. Or they exist in such extreme forms that life had to invent entirely new metabolic pathways, entirely new structural chemistry, entirely new sensory systems. And here's what we're only beginning to understand. These creatures aren't poorly adapted surface animals struggling to survive in a harsh environment. They're perfectly adapted deep-sea organisms for whom the surface would be a nightmare. The pressure we fear, they need it. The darkness we die in, it's their refuge. The cold, the scarcity, the isolation, those aren't obstacles. Those are features of the ecosystem they've mastered. We've been thinking about deep-sea life all wrong. We've been asking how do they survive down there? When we should be asking, what did the ocean allow life to become that the land never could? Because the ocean didn't just hide creatures from another world, it created another world. A world where evolution followed different physics, different chemistry, different biological logic. And tonight we're going to trace how it happened. How pressure reshaped cells, how darkness birthed light, how scarcity engineered patience, how isolation fostered the strangest biodiversity on Earth. We'll explore the chemistry that shouldn't work, the anatomy that defies structure, the senses that detect what can't be seen, the metabolisms that run on almost nothing. By the end, you might not look at the ocean the same way, because it's not just water, it's a laboratory, and for 400 million years, it's been running the most radical experiment in evolutionary biology Earth has ever seen. Let's begin with the simplest question. The one that breaks everything we thought we knew about life. How do you see in a place where light doesn't exist? The question seems simple. How do you see in a place where light doesn't exist? The answer evolution found is almost paradoxical. You make your own. Bioluminescence, the production of light by living organisms through chemical reactions, isn't rare in the ocean. It's not even uncommon. It's the dominant form of communication on the planet. More organisms speak in light than in sound, in scent, in touch. In the mesopelagic zone, the twilight layer between 600 and 3,300 feet deep, an estimated 76% of all visible organisms can produce their own light. Let that sink in. 3 out of every 4 creatures you'd encounter in the largest habitat on Earth glow. But here's what makes this extraordinary. Bioluminescence didn't evolve once and spread. It evolved independently, over and over again, in completely unrelated organisms solving the same problem in the same way. According to research published across multiple studies, bioluminescence has arisen at least 40 separate times across the tree of life, and likely more than 50 times when you account for recent discoveries. In 2016, researchers analysing ray-finned fishes alone found that bioluminescence evolved 27 independent times just within that group, 27 separate occasions where fish lineages, scattered across millions of years of evolution, looked at the darkness and invented light. This is convergent evolution on a scale that borders on inevitability. When the same solution appears dozens of times independently, you're no longer looking at accident. You're looking at physics and chemistry conspiring to make certain outcomes nearly mandatory. The chemistry itself is elegant. At its core, bioluminescence requires a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferin reacts with oxygen in the presence of luciferase, it releases energy, not as heat, but as cold light. It's one of the most efficient energy conversions in nature, with nearly 100% of the chemical energy transformed directly into photons. But here's where it gets strange. Luciferin doesn't vary much across species. One molecule in particular, calentrazine, serves as the light-emitting pigment for 9 completely different phyla. Radiolarians, comb jellies, cnidarians, crustaceans, mollusks, arrowworms and vertebrates all use the same basic chemical to glow. Yet luciferase, the enzyme that controls the reaction, varies wildly. Almost every species has its own unique version. It's as if evolution handed out the same flashlight bulb to dozens of unrelated organisms, but each one had to invent their own battery and switch. Some organisms synthesize calentrazine themselves, others obtain it through their diet, eating creatures that produce it and recycling the molecule for their own uses. It creates a glowing food web, where light compounds cascade through the ecosystem like nutrients, passed from prey to predator, each organism adding its own bioluminescent signature. And the purposes? They're as varied as the organisms themselves. Some use light to hunt. The anglerfish is famous for this, a glowing lure dangling from a modified dorsal spine, wiggling in the darkness like bait on a fishing line. Prey sees movement, approaches what they think is food, and find teeth instead. The anglerfish doesn't chase. It waits, motionless in the black water, letting curiosity do the work. But not all anglerfish lures work the same way. Different species have evolved wildly different light organs. Some have simple glowing bulbs. Others have elaborate branching structures filled with bioluminescent bacteria housed in specialized chambers. The flashlight fish, relatives of the anglerfish, have pockets beneath their eyes filled with luminous bacteria that they can cover and uncover with muscular shutters, creating blinking patterns like Morse code. According to a 2016 study in PLOS One, these bacterial partnerships have evolved at least 17 separate times in fish alone. The bacteria, mostly from the genus Vibrio, are essentially free-living organisms that fish capture from the environment and cultivate in specialized organs. The fish provide nutrients and protection, the bacteria provide light, it's symbiosis written in photons, and the fish control that light with remarkable precision. Ponyfishes, for instance, have evolved sexually dimorphic shutters and species-specific translucent windows to focus or broadcast bacterial light in patterns unique to each species. Deep-sea anglerfishes have sculpted their bacterial light organs into elaborate lures that vary so dramatically between species that you can identify them by their eskers alone. Other creatures use light for camouflage, which sounds counterintuitive until you understand the physics. In the twilight zone, faint sunlight still filters down from above. If you're a fish swimming in mid-water, you cast a silhouette, a dark shape against the dim glow from the surface. Predators lurking below can see that silhouette clearly. But if you have photophores, light-producing organ, on your underside that match the intensity and colour of the downwelling light, you erase your shadow. You become invisible. This is called counter-illumination, and it's so effective that dozens of species have independently evolved the same strategy. The hatchetfish, for example, has rows of photophores along its belly that it continuously adjusts to match the ambient light from above. Photoreceptors on its back measure incoming light. Neural circuits calculate the needed intensity. The photophores respond in real time, dimming and brightening to maintain perfect camouflage. It's active camouflage controlled by what is essentially a biological light meter and feedback loop, operating automatically in a fish brain the size of a grain of rice. Then there's defensive bioluminescence, and this is where things get creative. When attacked, some organisms don't run. They scream, not in sound, in light. They produce what researchers call a burglar alarm display, a sudden explosion of bioluminescence designed not to blind the predator, but to attract something bigger. The strategy is brilliant. If you're being eaten, light up as brightly as possible. Make yourself and your attacker visible. Anything larger and hungrier that sees the commotion will investigate, and suddenly your predator has become prey. You, in the chaos, slip away into the darkness. The vampire squid does a variation of this. When threatened, it ejects that sticky cloud of bioluminescent mucus, thousands of glowing particles that adhere to the predator's body, marking them like a beacon. According to observations from the Monterey Bay Aquarium Research Institute, this luminous cloud can persist for nearly 10 minutes, plenty of time for larger predators to home in on the glowing target. But perhaps the most sophisticated use of bioluminescence is communication, especially in finding mates. In the vast, dark ocean, how do you find another member of your species? Sound doesn't travel well at certain frequencies. Chemical signals disperse slowly. But light? Light can be seen from hundreds of feet away, and it can encode complex information in ways scent never could. Many bioluminescent species have evolved species-specific flash patterns, rhythms and intensities unique to each type. Firefly squid, for instance, gather into massive mating aggregations off the coast of Japan each spring, creating synchronized light shows that can be seen from shore. Each species flashes its own pattern, a visual dialect that ensures they only attract the right mates. Lanternfishes, the family Myctophidae with over 250 species, are perhaps the most diverse bioluminescent vertebrates. They're also among the most abundant fish in the ocean, and each species has a unique arrangement of photophores on its body. Pattern and position matter. Number of photophores matters. Sexual dimorphism exists. Males and females often have different light arrangements. According to a 2024 study in Nature, Ecology and Evolution, this diversity isn't accidental. Lanternfishes that evolved species-specific bioluminescent patterns diversified into new species at much faster rates than their relatives without them. The light patterns act as reproductive isolators. Fish that recognize each other's flash patterns mate preferentially, splitting populations even when they share the same habitat. Bioluminescence, in this case, doesn't just help fish find mates, it actively drives speciation. It's both a communication system and an evolutionary engine. And then there's red bioluminescence, which is vanishingly rare and utterly fascinating. Most deep-sea bioluminescence is blue-green. Blue wavelengths travel farthest through water, up to 100 meters in clear ocean water, compared to just 15 meters for red light. Red light is absorbed so quickly that by the time you're 50 feet deep, it's functionally extinct. This is why many deep-sea animals are red. In the absence of red light, red pigmentation renders them invisible, appearing black against the dark water. But a handful of species, notably dragonfish in the family Stomidae, have evolved the ability to produce and see red bioluminescence. According to research published in Science in 2005, these fish have a secret spotlight. They produce red light from photophores beneath their eyes, illuminating prey that other predators can't detect. It's like having infrared night vision in a world where everyone else is blind to that spectrum. They can see their food can't see them. Their competitors can't see what they're doing. The chemistry required for this is extraordinary. Producing red bioluminescence requires a different luciferin or modifications to the standard blue-green systems. The visual pigments needed to detect red light require specialized opsins tuned to longer wavelengths. These fish had to evolve both the sender and the receiver, the flashlight and the eyes to see by it. And they did it independently. At least 3 separate lineages of dragonfish evolved red bioluminescence, each using slightly different chemical mechanisms. Convergent evolution again, but this time on a private channel. What does all this tell us? That bioluminescence isn't an exotic adaptation. It's a fundamental reimagining of what light means. On land, light comes from the sun. In the deep ocean, light comes from life. It's not just vision. It's language, camouflage, weaponry, courtship, speciation. It's chemistry turned into communication, at scales we're only beginning to measure. But it raises a deeper question. If the absence of light drove the evolution of bioluminescence 40+ independent times, what else has the deep ocean forced life to reinvent? Because light is just the beginning. The darkness is only one challenge. There's also the pressure, and the pressure changes everything. At 10,000 feet below the surface, the pressure reaches 300 atmospheres. That's 4,400 pounds per square inch pressing down on every surface. To put that in human terms, imagine the entire NFL and NBA standing on your chest, all at once. That's what a fish experiences just existing at that depth. At 26,000 feet, the depth of the Mariana Trench, pressure exceeds 1,000 atmospheres, over 15,000 pounds per square inch. At this pressure, styrofoam cups shrink to the size of thimbles, air pockets in submersibles compress to dangerous levels. The human body, being mostly water, would fare slightly better than a submersible, but our insistence on keeping air in our lungs would kill us instantly. And yet, life exists there, not struggling, thriving. The Hadal snailfish, Pseudoleparus swirri, holds the record for the deepest living fish ever captured. Found at 26,131 feet in the Mariana Trench, this pale, translucent creature swims effortlessly in water that would crush almost anything from the surface. It has no scales, no swim bladder. Its bones are barely calcified, giving it a gelatinous, almost boneless structure. But here's the truly remarkable part. When brought to the surface, these fish die. Not from explosive decompression like you'd expect, but because their biochemistry literally stops working at normal pressure. They're not adapted to survive the deep. They're evolved to require it. The key to understanding this lies in a molecule with a distinctly unpleasant association, trimethylamine oxide, or TMAO. If you've ever smelled fish that's gone bad, you've smelled trimethylamine, the breakdown product of TMAO that gives rotting seafood its characteristic stench. But in living fish, TMAO serves a far more elegant purpose. It's a pressure counteractant, a molecular shield that protects proteins from being crushed and deformed by the immense forces of the deep. The story of TMAO begins with a simple observation. In the early 2000s, marine biologist Paul Yancey at Whitman College noticed something strange. Shallow water fish typically have TMAO concentrations around 40-70 millimoles per kilogram of muscle tissue. But when his team analysed deep-sea fish, grenadiers, rat-tails and other species living at 60 to 16,000 feet, the TMAO levels were dramatically higher. According to research Yancey published in 2014 in the Proceedings of the National Academy of Sciences, the pattern was striking. TMAO concentration increased linearly with depth. A fish at 3,000 feet might have 100 mg, at 6,000 feet 150 molu, at 13,000 feet over 200 mol/kL, and the Hadal snailfish from 26,000 feet, 261 mol/kL, the highest TMAO concentration ever recorded in any fish. This wasn't random. TMAO levels tracked depth with mathematical precision, increasing at roughly 20 millimoles per kilometre of descent. And of all the environmental variables that change with depth, temperature, light, oxygen, nutrients, only one changes in a perfectly linear way: pressure. But why would fish need more TMAO as pressure increases? The answer lies in what pressure does to proteins. Proteins are chains of amino acids folded into precise three-dimensional shapes. Those shapes determine function. An enzyme works because its active site has exactly the right geometry to bind substrates and catalyze reactions. A structural protein provides support because its folded configuration creates rigidity. Pressure destabilizes these folds. At high pressure, the forces between atoms shift. Hydrogen bonds weaken. Hydrophobic regions that normally cluster together spread apart. The protein begins to denature, not from heat, but from mechanical compression. For most proteins from surface organisms, pressures above 400 to 500 atmospheres cause catastrophic loss of function. Enzymes stop catalyzing. Structural proteins collapse. Muscle fibers fail to contract. Life, as designed for the surface, simply breaks. But TMAO changes the game. According to studies conducted by Yancey and marine biochemist Joseph Siebenhaler, TMAO acts as what's called a chemical chaperone. It doesn't bind directly to proteins. Instead, it alters the properties of the water surrounding them. Water under pressure becomes more ordered, more structured. This structured water penetrates into proteins, forcing open cavities and unfolding regions that should stay compact. But TMAO disrupts that process. It bonds with water molecules in ways that preserve the water's normal, less structured state, even under crushing pressure. The result? Proteins stay folded, enzymes keep working, muscle contracts, life continues. In laboratory experiments, researchers subjected lactate dehydrogenase, a critical enzyme in muscle metabolism, to 500 atmospheres of pressure. Without TMAO, the enzyme's activity dropped by 15%. Its ability to bind substrates degraded. At even higher pressures, it lost half its function. But when 250 millimoles of TMAO was added to the solution, the enzyme worked perfectly. At 500 atmospheres, equivalent to living at 16,000 feet deep, performance matched what you'd expect at the surface. The same held true for other proteins. Actin, the protein that forms muscle filaments, normally can't polymerize under high pressure. The pressure disrupts the assembly process, but add TMAO and actin polymerizes normally even at 500 atmospheres. According to a 2022 study published in Communications Chemistry, researchers used neutron scattering to observe exactly what TMAO does at the molecular level. They subjected water to 4,000 bar of pressure, roughly 40 times atmospheric pressure, both with and without TMAO present. Under pressure alone, the water's hydrogen bonding network collapsed. The second solvation shell, the layer of organized water molecules surrounding other water molecules, disappeared. This is the change that penetrates proteins and destroys their structure. But with TMAO present, that second solvation shell remained intact. TMAO formed strong hydrogen bonds with water molecules, creating a protective cage that resisted pressure-induced distortion. The water, even under extreme force, behaved almost as if it wasn't under pressure at all. And here's where it gets mathematically beautiful. The researchers calculated an osmolyte protection ratio, the concentration of TMAO needed to counteract a given amount of pressure. That ratio, derived from molecular physics, matched almost perfectly with the TMAO concentrations actually found in fish at corresponding depths. Physics predicts what evolution discovered, but there's a catch, a fundamental limit. Fish are osmoregulators, unlike invertebrates, which allow their internal osmolality to match seawater, roughly 100 milliosmoles per kilogram. Fish actively maintain lower internal osmolality, typically around 350 kilograms. They do this by excreting salt through their gills and kidney, maintaining an osmotic gradient that keeps water from constantly flooding their cells. But TMAO is an osmolyte. It contributes to internal osmotic pressure, and as TMAO concentrations rise with depth, so does the fish's total internal osmolality. At shallow depths, this isn't a problem. A fish with 50 mulk TMAO maintains an osmolality well below seawater, but a Hadal snailfish with 261 mulk TMAO pushes internal osmolality towards 700-800 multimosm. That's approaching the osmolality of seawater itself. Yancey's team did the math. If you extrapolate the linear increase of TMAO with depth, fish would reach isosmotic conditions, where their internal fluids match seawater, somewhere between 26,000 and 28,000 feet. Beyond that depth, fish would need more osmolality than seawater to continue producing enough TMAO. They'd have to reverse their osmotic gradient. salt would flow inward instead of outward. Their entire osmoregulatory system, evolved over hundreds of millions of years to pump salt out, would have to run backward. It's mechanically possible, but evolutionarily implausible. The energetic costs would be staggering. The cellular machinery would need complete redesign, and tellingly, no fish have ever been found below 27,000 feet. The deepest 25% of the ocean, from 27,000 to 36,000 feet, appears to be entirely fishless. This isn't speculation. Multiple deep-sea surveys using baited cameras, submersibles and traps have explored Hadal trenches worldwide. They find amphipods, sea cucumbers, snailfish down to about 27,000 feet. Below that, invertebrates continue, but fish disappear. TMAO may be the biochemical barrier that sets the depth limit for all fish. Not because pressure crushes them, but because the chemistry required to resist that pressure creates osmotic constraints they can't overcome. Life isn't prevented from going deeper by mechanical forces, it's constrained by the math of molecular protection. But this raises an even stranger question. If fish are biochemically locked out of the deepest trenches, what about organisms that don't have the same osmotic constraints? Invertebrates, particularly amphipods, have been found thriving at 36,000 feet, the very bottom of the Mariana Trench, and they use the same TMAO strategy. According to a 2018 study published in Deep Sea Research, amphipods from Hadal depths contain TMAO concentrations exceeding 300 mmol and even higher than the deepest fish. But amphipods are osmoconformers. They don't fight against seawater's osmolality, they match it. Their internal fluids are naturally around 100 massamosmkus and dominated by sodium and chloride. Adding TMAO doesn't create an osmotic problem because they're not trying to maintain a gradient. They can keep increasing TMAO indefinitely without hitting the wall fish encounter. So the deepest parts of the ocean aren't lifeless, they're just reserved for organisms with different osmotic strategies. Fish evolved in a way that brilliantly solved mid-depth survival, but accidentally created a ceiling they can't break through. Invertebrates, following a different evolutionary path, face no such limit. It's a stunning example of how evolution's solutions aren't perfect. They're constrained by prior commitments. Fish became osmoregulators hundreds of millions of years ago, and that decision, made in shallow seas, ultimately determines how deep their descendants can ever go. But TMAO isn't the only pressure adaptation, it's just the most elegant chemical one. Other organisms have found different solutions. Some bacteria from Hadal depths have evolved pressure-activated enzymes, proteins that actually work better under compression. Their optimal activity occurs at 400–800 atmospheres. At surface pressure, they're sluggish, inefficient. Take them to the deep, and they thrive. According to research on pisophilic, pressure-loving bacteria published in Frontiers in Microbiology, some species have even evolved pressure-dependent gene regulation. Certain genes only activate above 200 atmospheres. They've wired pressure into their genetic circuitry as an environmental signal, the way surface organisms use temperature or light. And then there are the structural adaptations that seem almost paradoxical. Deep-sea fish often have gelatinous, watery tissues, up to 95% water in some species. Their bones are poorly mineralized, sometimes barely present. They have minimal musculature. This isn't weakness, it's strategy. Water is incompressible. Unlike air, which collapses under pressure, water maintains its volume. A body filled with water doesn't get crushed because there's nothing to compress. The pressure outside equals the pressure inside and everything equilibrates. But proteins, lipids, and complex molecules can still deform, so deep-sea organisms maximize water content and minimize the structural components that pressure can damage. They become, essentially, organized bags of seawater with just enough biochemistry to stay alive. The blobfish, made famous by photos showing its gelatinous, sad-looking face, is the perfect example. At depth, under pressure, the blobfish looks relatively normal. Its tissues, filled with low-density gel, maintain structure because the internal and external pressures balance, but bring it to the surface and it collapses. The pressure that was supporting its tissues vanishes. What was once firm at 3,000 feet becomes a shapeless blob at sea level. The blobfish didn't evolve to look sad. It evolved to look normal at depth, and we only see it out of context. Similarly, many deep-sea creatures have lost swim bladders. gas-filled organs that provide buoyancy in shallow fish. A swim bladder at 10,000 feet would compress to a fraction of its normal size, becoming useless or even dangerous. So deep-sea fish evolved different buoyancy strategies: oily livers, gelatinous tissues, reduced skeletal mass. They achieve neutral buoyancy through chemistry, not pneumatics. And their movement strategies change too. Muscle requires energy. At great depths, where food is scarce and metabolism runs slow, fast swimming is prohibitively expensive. So many deep-sea predators evolved ambush strategies. They hover, they drift, they wait with minimal movement, conserving every calorie until prey wanders close enough to strike. The gulper eel exemplifies this. Its body is essentially a giant mouth attached to a thin whip-like tail. It can unhinge its jaws to swallow prey larger than itself, and its stomach expands to accommodate huge meals. Because food is so rare at 6,000 feet, it has evolved to eat anything it encounters, regardless of size, then digest slowly over weeks. This is pressure's second effect. It doesn't just crush, it isolates. Food sinks slowly, nutrients disperse. The deep ocean is a desert, and pressure is part of why. Pressure makes it energetically expensive to move, to hunt, to maintain fast metabolisms, so life slows down, and in slowing down it becomes patient in ways surface organisms never achieve. The Greenland shark, which hunts in deep, cold Arctic waters, at pressures exceeding 200 atmospheres, has a metabolism so slow that it grows less than a centimetre per year. It doesn't reach sexual maturity until age 150. Some individuals may live over 400 years, the longest-lived vertebrates on Earth. That's not just adaptation to cold, it's adaptation to an entire suite of deep-sea conditions where pressure, temperature, darkness, and scarcity conspire to make speed and aggression wasteful. Patience, in the deep, is survival. What does all this mean? It means pressure didn't just shape what organisms look like, it shaped how they think, how they move, how they allocate energy, how fast they live. It created biochemical barriers that entire groups can't cross. It forced life to invent molecules like TMAO that we're only now understanding at the atomic level. And it reveals something profound. The deep ocean isn't Earth with more water on top. It's a fundamentally different physical regime, where the rules of biochemistry, physiology, and ecology all had to be rewritten. But pressure is only half the equation. There's something else the deep ocean lacks, something surface life takes for granted, something so fundamental that its absence should make complex life impossible. Oxygen. You could drown in oxygen-rich water. Your lungs would fill, and you'd suffocate. But imagine drowning in water that itself is suffocating. Water so depleted of oxygen that even fish can't breathe. These zones exist. They're called oxygen minimum zones, or OMZs, and they occupy massive volumes of the ocean, primarily at depths between 600 and 3,000 feet. In some regions, off the coast of California, in the eastern tropical Pacific, the Arabian Sea, oxygen concentrations drop to less than 0.5% of surface levels. To put that in perspective, surface seawater at equilibrium with the atmosphere contains roughly 200 250 micromoles of oxygen per litre. In oxygen, minimum zones, concentrations can plummet below 20 micromoles per litre. In the most extreme cases, in what researchers now call anoxic marine zones, oxygen becomes undetectable even with the most sensitive instruments. Less than 5 micromoles per litre, essentially zero. And yet, life exists there, not barely clinging to survival, thriving, abundant. Some of the ocean's richest ecosystems exist in these suffocating layers. The question isn't whether life can survive low oxygen. The question is how it's possible at all. The answer involves some of the most radical biochemical engineering evolution has ever produced. First, understand what oxygen minimum zones are and why they form. They're not random. They're predictable consequences of ocean physics and biology working together. Start at the surface. Sunlight drives photosynthesis. Phytoplankton, microscopic algae drifting in the water column, produce oxygen as a byproduct. Surface waters stay well oxygenated, constantly mixing with the atmosphere, saturated with dissolved gases. But phytoplankton don't live forever. They die, they sink, and as they sink, they become food. Bacteria in the water column feast on this marine snow, a constant rain of dead plankton, fecal pellets, shed scales, mucus, organic debris. And bacteria breathe. They consume oxygen to metabolize organic matter, breaking it down into nutrients and carbon dioxide. The deeper you go, the more accumulated detritus there is to consume, and crucially, the less mixing occurs with oxygen-rich surface water. Deep water moves slowly, sometimes taking decades to circulate back to the surface. Once the oxygen in that water is consumed by microbial respiration, it can't be replaced without the water mass reaching the surface again. In certain regions, particularly where nutrient-rich upwelling drives massive phytoplankton blooms, creating huge amounts of sinking organic matter, microbial respiration consumes oxygen faster than circulation can replenish it. The result, an oxygen minimum zone, a layer of water, often hundreds of meters thick, where dissolved oxygen drops to levels that would kill most surface-adapted organisms in minutes. According to a study published in the Journal of Experimental Biology in 1998 by marine physiologists James Childress and Brad Seibel, animals living in oxygen minimum zones off the coast of California face partial pressures as low as 0.8 kilopascals, about 6 torr, or less than 1% of atmospheric pressure. To a mammal, that's lethal. You'd lose consciousness within seconds. But the animals there have solved this problem through a suite of adaptations so complete, so integrated, that they've essentially re-engineered how respiration works. Start with extraction efficiency. Most fish extract about 50–60% of the oxygen from water, passing over their gills. That's respectable. But in oxygen minimum zones, that's nowhere near enough. Species like the bathypelagic mysid Gnathopausia ingens, a shrimp-like crustacean that lives at depths where oxygen drops to 6 torr, can extract up to 90% of available oxygen from ventilated water. How? Massive gills with enormous surface area. According to research published in the Biological Bulletin in 1990 by Childress and colleagues, these animals have evolved gill structures with exceptionally thin diffusion barriers, sometimes just a few micrometers separating water from blood, and gill surface areas far exceeding what you'd find in surface-dwelling relatives. But extraction is only half the problem. You also need transport. This is where hemocyanin becomes critical. Most vertebrates use hemoglobin, an iron-based protein that turns blood red, to transport oxygen. Hemoglobin works brilliantly at normal oxygen levels, but many deep-sea invertebrates, including crustaceans and cephalopods, use hemocyanin instead, a copper-based protein that turns blood blue. And in oxygen minimum zones, hemocyanin has a crucial advantage. It can be tuned to have extraordinarily high oxygen affinity. Gnathopausia ingens has hemocyanin with a P50, the oxygen partial pressure at which the protein is 50% saturated, of just 1.4 torr at physiological temperature and pH. That means this animal's blood can load oxygen even when environmental levels drop to barely detectable amounts. For comparison, human hemoglobin has a P50 around 27 torr. At 6 torr, your hemoglobin would be less than 25% saturated. You'd be hypoxic, approaching organ failure. But Gnathopausia's hemocyanin would still be over 80% saturated, fully functional. But here's the engineering challenge. If your blood protein binds oxygen too tightly, it won't release it at the tissues. High affinity is useless if you can't deliver. Deep-sea animals solve this with cooperativity and the Bohr effect. Cooperativity means that once one oxygen molecule binds to hemocyanin, the protein changes shape slightly, making it easier for additional oxygen molecules to bind. This creates a steep binding curve. Oxygen loads rapidly when available, but unloads rapidly when partial pressure drops slightly. Gnathopausia's hemocyanin has a cooperativity coefficient, N50, of 3.5, exceptionally high for a crustacean. This means that across a very small change in oxygen pressure, say from 6 torr in the gills to 4 torr in the tissues, the hemocyanin goes from nearly fully loaded to substantially unloaded, delivering oxygen efficiently despite the tiny gradient. And then there's the Bohr effect, the tendency for respiratory proteins to release oxygen when pH drops. In metabolically active tissues, cells produce carbon dioxide, which lowers local pH. This acidification causes hemocyanin to release oxygen right where it's needed most. Gnathopausia has one of the largest Bohr effects ever measured in a crustacean respiratory protein, a log P50 dpH of 0.80. That means a small drop in pH causes a massive increase in oxygen release. Put it all together. Massive gills extract 90% of available oxygen. High-affinity hemocyanin loads that oxygen even at 1,2 Tori. High cooperativity and a large bore effect ensure efficient delivery to tissues across minimal gradients. It's biochemical perfection, and it's not unique. The vampire squid, living in even more extreme oxygen minimum zones, where levels drop below 5 micromoles per litre, has evolved similar strategies. Its hemocyanin binds oxygen so efficiently that the animal can extract enough to support aerobic metabolism in water that would asphyxiate almost any other active predator. According to research published in Progress in Oceanography in 2010, the vampire squid's closest relatives, other omastrophid squids like Ilex illicabrosus, have hemocyanin P50 values around 8-10 Tora. They're adapted to well-oxygenated Atlantic waters. But the vampire squid, its P50 drops below 2 Tori, and remarkably, that affinity changes with temperature and depth. When the squid is in cold, deep, oxygen-depleted water, its hemocyanin affinity increases. When it occasionally ventures into warmer, oxygen-rich surface waters, affinity decreases, facilitating faster oxygen release to active tissues. It's dynamic regulation. The same protein functions differently depending on environmental conditions, tuned by temperature and pH to optimize performance across wildly varying oxygen landscapes. But even the most efficient oxygen extraction and transport can't support high activity levels when oxygen is scarce. So many oxygen minimum zone residents have adopted a different strategy: metabolic suppression. The Humboldt squid, Dosidicus gigas, is a massive active predator that hunts in the oxygen minimum zones of the eastern Pacific. These squid can reach 6 feet in length and weigh over 100 pounds. They're aggressive, fast-swimming carnivores with metabolic demands that should be incompatible with low-oxygen environments, and yet, they spend significant portions of their day in the oxygen minimum zone, at depths where oxygen concentrations drop below 10 micromoles per litre. How? According to studies conducted by Seibel and published in 2011 in the Journal of Experimental Biology, Humboldt squid suppress their metabolism by up to 40% when they enter low-oxygen waters. They don't shut down completely. They remain alert, capable of predation, but they dramatically reduce energy expenditure. Their routine metabolic rate, the energy needed just to stay alive and move slowly, drops from roughly 50 micromoles of oxygen consumed per gram per hour at the surface to around 30 micromoles per gram per hour in the oxygen minimum zone. This isn't passive drowsiness, it's active metabolic regulation. The squid reduce gill ventilation frequency. They slow movement. They may even reduce neural activity in non-essential systems. And they switch partially to anaerobic metabolism, producing lactate as a byproduct of glycolysis when aerobic ATP production can't meet demands. But anaerobic metabolism is expensive and unsustainable. Lactate accumulates. It creates acid. It has to be cleared eventually, and clearing it requires oxygen. So the Humboldt squid uses anaerobic pathways only as a supplement, covering maybe 10-20% of their energy needs during hypoxic forays. The rest comes from ruthlessly efficient aerobic metabolism running at minimal intensity. And here's the fascinating part. This strategy is reversible. When the Humboldt squid ascends at night into oxygen-rich surface waters to hunt, its metabolism ramps back up. Heart rate increases, ventilation frequency increases. It pays back the oxygen debt accumulated during the day, clearing lactate, restoring ATP reserves. The squid essentially commutes between two metabolic states, suppressed and aerobic in the daytime oxygen minimum zone, fully active and aerobic at night in surface waters. It's a daily metabolic migration as dramatic as its vertical migration through the water column. Other species don't migrate. They live permanently in the oxygen minimum zone, and for them, metabolic suppression isn't temporary, it's a way of life. Deep-sea fishes that inhabit oxygen minimum zones have routine metabolic rates 10 to 20% of what you'd find in similarly sized shallow water species. They move slowly, they grow slowly, they reproduce infrequently. Everything is paced to minimize oxygen consumption. But don't mistake this for weakness. These fish aren't struggling, they're optimized. In an environment where food is scarce and oxygen is limited, A slow metabolism is an advantage. It means longer survival between meals. It means less vulnerability to brief periods of even lower oxygen. It means energy can be allocated to reproduction, growth, and immune function, instead of wasted on unnecessary activity. And the evolutionary results are striking. According to a 2011 study by Seibel, organisms living permanently in oxygen minimum zones have critical oxygen levels, the picoreate, below which they can no longer maintain aerobic metabolism, as low as 1 to 2 torr. Compare that to surface species, which typically have picoreate values of 40-80 torr, and the adaptation becomes clear. An organism with a picoreate of 2 torr can remain fully aerobic in water where oxygen is barely detectable. It's not gasping, it's not distressed, it's functioning normally in conditions that would be lethal to its ancestors. But there's a limit to even these adaptations. In the most extreme oxygen minimum zones, the anoxic marine zones where oxygen drops to 0, even the most efficient aerobic metabolism fails. And here, life faces a choice: leave or switch to chemistry that doesn't require oxygen at all. Some bacteria make that switch. They use nitrate instead of oxygen as an electron acceptor, conducting denitrification, a form of anaerobic respiration that converts nitrate to nitrogen gas. Vast bacterial communities thrive in anoxic marine zones, cycling nitrogen through pathways that bypass oxygen entirely. These microbes can dominate the ecosystem. In the oxygen minimum zone off the coast of South America, researchers have found bacterial mats the size of Uruguay floating in the water column, processing nutrients, consuming organic matter, and fundamentally altering ocean chemistry on a continental scale. But complex animals, fish, squid, crustaceans, can't sustain themselves on anaerobic metabolism alone. The energy yield is too low, the waste products too toxic. So the true anoxic zones, where oxygen reaches 0 and stays 0, are largely devoid of complex metazoans. They're bacterial worlds. Which brings us to a profound realization. Oxygen minimum zones aren't just challenging environments that a few hardy species tolerate. They're selective filters that have driven some of the most sophisticated respiratory and metabolic adaptations in the animal kingdom. The vampire squid's hemocyanin, the Humboldt squid's metabolic suppression, Gnathopasia's massive gills and cooperative oxygen binding. These aren't minor tweaks. They're comprehensive re-engineering of how energy is acquired and used. And they reveal something crucial. The deep ocean didn't just remove light and add pressure. It also removed the oxygen we take for granted. And in doing so, it forced life to invent respiratory strategies that don't exist on land, metabolic flexibilities surface organisms never needed, biochemical solutions to problems that never arose in well-oxygenated ecosystems. The ocean minimum zones are laboratories of physiological innovation, and the creatures that live there are proof that impossible conditions just mean evolution hasn't had enough time yet. But even metabolic suppression, high affinity hemocyanin, and massive gills can't explain everything we see in the deep. Because some adaptations aren't about extracting more from less. They're about sensing what shouldn't be sensible, detecting signals in a world where conventional senses fail. In the darkness and scarcity of the deep, survival isn't just about breathing. It's about knowing what's out there, even when you can't see, can't hear, and can barely detect anything at all. In complete darkness, under crushing pressure, with minimal oxygen and almost no sound, how do you find food? How do you avoid becoming food? How do you find a mate in three-dimensional space when you can't see more than a few feet in any direction? The answer: you stop relying on the senses that work on land, and you develop entirely new ways of perceiving the world. Deep-sea creatures don't just have better versions of human senses, they have senses we don't possess at all. Senses that detect things we can't even imagine detecting. Senses that turn the ocean into a readable landscape of information invisible to us. Start with the lateral line system, perhaps the most alien sensory organ fish possess, and one we have no equivalent for. Every fish, from shallow water minnows to hadal zone snailfish, has a lateral line running along each side of its body. If you look closely at a fish, you'll see it, a faint line of small pores running from head to tail. Those pores aren't decorative. They're openings to a sophisticated mechanoreceptive network that allows fish to feel water movement at a distance. Inside those canals, embedded in the skin, are specialized sensory structures called neuromasts. Each neuromast consists of hair cells, the same type of mechanoreceptive cells found in your inner ear, topped with a gelatinous cupula that acts like a fluid-filled dome. When water moves across the fish's body, it creates pressure differentials. Water flows through the pores, creating currents in the lateral line canals. Those currents bend the cupulae, which deflect the hair cells beneath them. The hair cells depolarize, sending electrical signals to the brain. The result? The fish can detect vibrations, pressure waves, and water displacement from meters away. It can sense the movement of nearby objects without seeing, hearing, or touching them. It can feel the wake left by another fish that passed by seconds ago, tracking prey or predators through invisible trails in the water. According to research on the lateral line system, fish use this sense for everything: navigating in darkness, maintaining position in schools, detecting obstacles, locating prey, and avoiding predators. In species that live in caves, or the deep sea where vision is useless, the lateral line becomes the primary sensory system. And in the deep sea, where every photon of bioluminescent light is precious and revealing yourself visually could mean becoming prey, the lateral line offers something vision never could: passive detection. You can sense without being sensed. You can track without emitting. You leave no signature except the disturbance you make moving through water, which if you move slowly enough becomes nearly undetectable. Deep-sea fish have taken the lateral line to extremes. Some species have expanded canal systems covering not just the body trunk but the entire head, with branches extending across the skull, around the eyes, along the jaw. The density of neuromasts in these canals far exceeds what you find in shallow water fish. Why? Because in the deep sea, the lateral line isn't supplementing vision, it's replacing it. But the lateral line, as sophisticated as it is, can only detect movement and pressure. It can't detect something that's perfectly still, and it definitely can't detect something buried in sediment. For that you need electroreception. Electroreception, the ability to sense electrical fields, is perhaps the most alien sense to humans. We have no electroreceptive organs. We can't detect bioelectric fields at all. But for many marine animals, particularly sharks, rays, and some deep-sea fish, electroreception is as fundamental as vision is to us. Every living cell generates electrical potential. When muscles contract, neurons fire, or hearts beat. They create tiny electrical fields that propagate through the surrounding medium. In air, these fields dissipate almost instantly, but water, especially seawater with its dissolved salts, conducts electricity remarkably well. Bioelectric fields can extend several feet from their source, and some animals can detect fields as weak as 5 nanovolts per centimetre. That's 5 billionths of a volt. To put that in perspective, if you place 2A batteries 3,000 miles apart in the ocean, a shark could theoretically detect the voltage gradient between them. This extraordinary sensitivity comes from specialised organs called ampullae of Lorenzini, most famous in sharks but present in rays, skates, chimeras, and even some bony fish like paddlefish and sturgeon. Ampullae of Lorenzini appear as small pores on the skin, concentrated mainly on the head. Each pore opens into a canal, sometimes over a foot long, filled with conductive jelly. At the end of each canal sits a cluster of sensory cells, hair cells similar to those in the lateral line but modified to detect voltage instead of pressure. According to research published in scientific reports in 2025 on deep-sea ghost sharks and spookfish, these animals possess extensive ampullary fields, covering their heads and snouts, with hundreds or even thousands of individual pores. The sheer density suggests these fish rely heavily on electroreception for hunting, and the strategy makes perfect sense. Most prey organisms, fish, crustaceans, worms, can't stop their hearts, can't prevent their muscles from generating bioelectric fields, can't shut down the electrical activity of their nervous systems, even if they're motionless, even if they're buried under sand, even if they're perfectly camouflaged, they're broadcasting their location electromagnetically. A predator with electroreception can find them anyway. Hammerhead sharks, with their bizarre flattened heads, may have evolved that shape specifically to improve electroreception. The wide, flat head increases the surface area available for ampullae, creating a broader electroreceptive antenna that can scan larger volumes of water and pinpoint prey location with greater precision. Experiments have shown that sharks can detect the heartbeat of a flatfish buried completely under sand. They swim over the seafloor, sweeping their heads back and forth like metal detectors, homing in on the rhythmic electrical pulses of a hidden heart. When they find it, they strike, accurately without ever seeing the prey. But electroreception isn't just for hunting, it's also for navigation. Earth itself generates electromagnetic fields. The planet's magnetic field creates predictable voltage gradients in seawater as ocean currents flow through it. Sharks and rays can detect these gradients. They use them as a navigational compass, orienting themselves relative to Earth's magnetic field, potentially migrating thousands of miles using electromagnetic cues we can't perceive at all. In the deep sea, where there are no visual landmarks, no sunlight to indicate direction, no stars to navigate by, electromagnetic navigation becomes crucial, and electroreception, evolved primarily for detecting prey, doubles as a geomagnetic sense. But both the lateral line and electroreception require external stimuli. They're passive senses, waiting for prey to move or hearts to beat. Some deep-sea creatures have evolved active sensing instead. They don't wait for signals, they create them. This is echolocation, and while we typically associate it with whales and dolphins, some deep-sea fish may use it too, though the evidence remains debated. Echolocation works by emitting sound and listening for echoes. The time delay between emission and return tells you distance. The echo's characteristics tell you size, shape, texture. It's biological sonar, and it works in total darkness. Toothed whales, sperm whales, orcas, dolphins use echolocation constantly, producing clicks and listening to how those clicks bounce off objects. Sperm whales hunt giant squid at depths exceeding 6,000 feet in absolute darkness, using only sound to locate prey the size of school buses in open water. According to research on sperm whale acoustic behaviour, they produce some of the loudest biological sounds ever measured, clicks reaching 230 decibels, louder than a jet engine. These clicks can travel miles through water, and the returning echoes provide enough information for the whale to construct a mental map of its surroundings. But fish don't have the anatomical equipment for echolocation that whales possess. They lack the specialized organs, the massive sound-producing structures, the sophisticated auditory processing centers. Most fish hear reasonably well, but they don't echolocate, except maybe some do. There's tantalizing evidence that certain deep-sea fish produce sounds and may use the returning reflections for spatial awareness. Sturgeons, lanternfish, some grenadiers, all have been recorded producing clicks, pops or drumming sounds. Whether these sounds serve echolocation or are simply communication signals remains unclear. What is clear is that deep-sea creatures have evolved to maximize every possible sensory input, because in an environment this hostile, information is survival. And then there's chemical sensing, which might seem mundane compared to electroreception and lateral lines, but in the deep sea becomes extraordinarily refined. Smell and taste, chemoreception, work differently underwater than in air. On land, we inhale air carrying volatile molecules, and olfactory receptors in our noses detect them. Taste requires direct contact, molecules dissolved in saliva touching taste buds. But underwater, everything is already dissolved. The distinction between smell and taste blurs. Fish have chemoreceptors distributed across their bodies, in nostrils, in the mouth, on fins, even on barbels, whisker-like appendages extending from the head. Deep-sea fish often have enormous olfactory organs relative to their body size. Some species have olfactory bulbs, the brain region processing smell, that dwarf the rest of the brain. Smell becomes the dominant sense. Why? Because chemical signals persist. A drop of blood, a trace of amino acids from decaying flesh, molecules sloughed off by living tissue. These substances diffuse slowly through water, creating plumes that can extend hundreds of meters. A fish with sensitive chemoreceptors can follow those plumes to their source, tracking prey or scavenging opportunities across vast distances. Hagfish, primitive, eel-like scavengers that inhabit the deep-sea floor, are perhaps the best example. Hagfish are nearly blind. They have no lateral line system. Their hearing is poor, but they have exquisitely sensitive chemoreceptors that can detect a single drop of fish oil diluted into millions of gallons of water. When a whale carcass falls to the deep sea floor, whale fall, one of the most nutrient-rich events in the deep ocean, hagfish can smell it from miles away. They converge on the carcass within hours, sometimes arriving in such numbers that the seafloor around the whale becomes carpeted with writhing bodies, all feeding simultaneously. They don't need to see the food. They smell their way to it through kilometers of dark water. But chemoreception isn't just for finding food, it's also for reproduction. Many deep-sea species exist at such low population densities that encounters between males and females are vanishingly rare. In the largest habitat on Earth, with individuals scattered across millions of cubic miles of water, finding a mate is statistically almost impossible, so females broadcast. They release pheromones, chemical signals that advertise reproductive readiness. Males, sometimes equipped with larger olfactory organs than females, detect these pheromones and follow the chemical gradient to its source. In some deep-sea anglerfishes, this strategy reaches an extreme. Males are tiny, sometimes 1/10 the size of females, and their entire existence revolves around finding a female. Once they detect a female's pheromones, they pursue relentlessly. Upon contact, the male bites the female and never lets go. His tissues fuse with hers. His circulatory system connects to hers. Over time, he degenerates, losing eyes, fins, most internal organs, until he becomes essentially a parasitic sperm-producing appendage attached to the female's body. She carries him with her, and when she's ready to spawn, his sperm is right there, permanently available. It's sexual parasitism enabled by chemoreception. The male found the female in the vastness of the deep ocean by smell alone, then ensured he'd never lose her. But even with all these senses, lateral lines detecting pressure waves, ampullae sensing bioelectric fields, chemoreceptors tracking molecular trails. There's one sensory challenge the deep sea creates that seems insurmountable. How do you use vision in a place where light doesn't exist? You'd think deep sea creatures would be blind. Many are. But others have evolved eyes more sophisticated, more sensitive, more specialized than almost anything found in shallow water. Deep sea fish eyes come in bizarre forms. Some species have tubular eyes shaped like binoculars, pointed upward, maximizing light collection from the faint bioluminescence above. The barreleye fish we discussed earlier exemplifies this. Eyes embedded in a transparent head, rotating inside their sockets, collecting every photon available. Other species have enormous eyes relative to their body size. The giant squid has eyes the size of dinner plates, the largest eyes in the animal kingdom. Not because it lives in well-lit water, but because massive lenses can collect more of the scarce light available at depth. And the retinas of deep-sea fish are packed with rod cells, the photoreceptors specialized for low-light vision. Some species have retinas composed almost entirely of rods, with no cone cells at all. They've sacrificed color vision entirely in exchange for maximum sensitivity to dim light. According to research on deep-sea fish vision, some species have evolved rhodopsin, the light-sensitive pigment in rod cells, with altered spectral sensitivities tuned specifically to detect bioluminescence at the wavelengths most common in their habitat. Their eyes aren't just sensitive, they're precisely calibrated to see the kind of light their prey, predators, and potential mates produce. But perhaps the strangest visual adaptation is found in species with multiple visual pigments in the same retina, not for color vision, but for detecting different intensities of bioluminescence, or distinguishing bioluminescent light from residual sunlight at twilight depths. The stoplight loosejaw dragonfish, which produces red bioluminescence, has eyes that can see red light. Rare in deep-sea fish, since red wavelengths don't penetrate deep water. This gives the dragonfish a private channel. It can illuminate prey with red light that only it can see, hunting with a spotlight invisible to everyone else. And then there are fish with accessory retinas, extra light collecting structures behind the main retina that reflect light back through the photoreceptors for a second pass, doubling the chance of photon detection. It's biological image amplification, like night vision goggles built into the eye. What all these adaptations reveal is that the deep sea isn't truly dark. It's dim, extremely dim, but dim enough that evolution could work with it, refining vision to absurd extremes, extracting information from almost nothing. But when vision fails, when light is truly absent, deep-sea creatures still navigate, still find food, still reproduce. Because they've built sensory systems that don't depend on light at all. They sense pressure waves through lateral lines. They detect heartbeats through electroreception. They smell molecules through chemoreceptors. They feel vibrations through mechanosensors distributed across their skin. They use statocysts, balance organs filled with calcium carbonate crystals, to maintain orientation in three-dimensional space. They've turned the ocean into a readable environment without needing to see it. And what this means is profound. These creatures aren't handicapped by darkness. They're not limited by the absence of light. They've simply moved to different information channels. They're perceiving a version of reality we can't access, building mental maps of their world from electrical fields, pressure gradients, and chemical trails. To them, the deep ocean isn't a sensory void. It's dense with information. It's a landscape as rich and detailed as any rainforest or coral reef, just written in a language we don't speak. And that raises a final, unsettling question. What else is out there that we can't detect? Not because it doesn't exist, but because our senses aren't tuned to perceive it. The deep ocean taught evolution that the senses we take for granted, vision, hearing, smell as we experience them, are just a narrow slice of available reality. There are fields, gradients, vibrations, signatures all around us that we're blind to. Deep-sea creatures see them, feel them, read them like text, and they remind us that reality isn't what exists, it's what we're equipped to perceive, which means the ocean might be even stranger than we think. Because if our technology can reveal worlds invisible to us, what might exist that even our technology can't detect yet? The deep ocean keeps its secrets, not because they're hidden, but because we're still learning how to look. There's a woodlouse, a pillbug, the little armoured crustacean you find under rocks in your garden, living in the deep sea that can grow to 30 inches long. 30 inches, that's the length of a house cat. It's called the giant isopod, Bathynomus giganteus, and it's one of the most dramatic examples of a phenomenon called deep-sea gigantism, the tendency for creatures living in the abyss to grow far larger than their shallow water or terrestrial relatives. A typical woodlouse is maybe half an inch long. The giant isopod is 60 times that size. 60 times? That's not adaptation, that's reinvention, and it's not alone. The giant squid reaches lengths over 40 feet. The colossal squid, even larger, may exceed 45 feet and weigh over 1,500 pounds. Japanese spider crabs have leg spans reaching 12 feet. Giant amphipods, shrimp-like crustaceans, can grow to the size of a human hand, when their shallow water cousins are barely a millimetre long. Giant tube worms stretch nearly 8 feet. Giant oarfish reach 30 feet. The list goes on. Why? What is it about the deep ocean that doesn't just allow gigantism, but seems to encourage it? The answer lies in a combination of physics, metabolism, and evolutionary pressure that makes the deep sea one of the few places on Earth where bigger is almost always better. Start with buoyancy. On land, size is constrained by gravity. The largest land animals, elephants, giraffes, are at the physical limits of what bone and muscle can support. Go bigger, and skeletal systems fail. Legs break. Hearts struggle to pump blood against gravity. Movement becomes impossible. But water changes everything. Water provides buoyancy. An organism's weight is counteracted by the water it displaces. In the ocean, you don't need massive skeletons to support body weight. You don't need powerful legs to stand. Structural support comes from water pressure equalizing inside and outside the body. This is why blue whales, the largest animals ever to exist, larger than any dinosaur, can only exist in water. On land, a blue whale would collapse under its own weight, its organs crushed, its skeleton inadequate. But in water, it floats effortlessly, 200 tons moving with grace through three-dimensional space. The same principle applies to deep-sea invertebrates. A giant isopod doesn't need thick armor or powerful muscles to support its enormous body. The water does that. Its exoskeleton needs only enough rigidity to protect organs and provide attachment points for muscles, The rest is gelatinous tissue, minimally structured, held in shape by internal and external pressure equilibrium. But buoyancy alone doesn't explain gigantism. Shallow water species have buoyancy too, and they stay small, so there must be other factors. Enter Kleiber's law, one of the most fundamental scaling principles in biology. Kleiber's law states that an animal's metabolic rate doesn't scale linearly with mass. It scales to roughly the 3/4 power of mass. In simpler terms, if you double an animal's weight, its metabolic rate doesn't double. It only increases by about 75%. Why? It comes down to surface area, volume, and the geometry of circulatory and respiratory systems. Metabolism generates heat. Heat must be dissipated through the skin. But surface area increases as the square of linear dimensions, while volume increases as the cube. Bigger animals have proportionally less surface area per unit mass, so they lose heat more slowly. To avoid overheating, they've evolved lower mass-specific metabolic rates. Additionally, the circulatory system, which delivers oxygen and nutrients, operates more efficiently at larger scales. Blood vessels, airways, and transport systems don't need to scale one-to-one with body size. A fractal branching network can service a large body with proportionally less metabolic overhead than servicing many small bodies with the same total mass. The upshot: large animals are metabolically more efficient. They need less food per gram of body weight than small animals. According to Kleiber's law, a cat weighing 100 times more than a mouse requires only about 32 times as many calories, not 100 times, just 32. In an environment where food is abundant, this doesn't matter much. But in the deep sea, where food is desperately scarce, metabolic efficiency becomes survival. The deep ocean is a desert. Sunlight doesn't penetrate, photosynthesis doesn't occur. The only food comes from above. Marine snow, the slow rain of organic debris drifting down from surface waters, dead plankton, fecal pellets, molted exoskeletons, fragments of jellyfish. Occasionally, a large carcass, a whale, a shark, a tuna, falls from above, but these events are rare, unpredictable, separated by years or decades. For a deep-sea scavenger, meals are sparse and sporadic. You might go months without encountering food. When you do find it, you need to consume as much as possible, storing it for the long famine ahead. This is where Kleiber's law becomes crucial. A large animal can survive longer between meals because it burns less energy per gram of tissue. It can store more reserves relative to its maintenance costs, and when food appears, it can consume more in a single feeding event. Giant isopods exemplify this. According to research on their feeding behaviour, they can gorge themselves to the point of near immobility, distending their bodies until they can barely move, and then they wait. Studies have shown that giant isopods in captivity can survive over 5 years without food. 5 years. That's not starvation resistance in the sense we usually think of it. That's a fundamentally different metabolic strategy. One where the baseline energy expenditure is so low that stored reserves can last half a decade. And it's only possible at large body sizes. A tiny isopod, with its higher mass-specific metabolic rate, would starve in months. But scale up to 30 inches, and suddenly 5 years of fasting becomes feasible. So buoyancy enables large size, and Kleiber's law makes large size advantageous in food-scarce environments. But there's a third factor: temperature. This is where Bergmann's rule comes in. The observation that animals in colder climates tend to be larger than their warm climate relatives. The principle is straightforward. Larger animals have lower surface area to volume ratios, so they lose heat more slowly. For warm-blooded animals trying to maintain body temperature, larger size conserves warmth, but the deep ocean is cold, near freezing, and most deep-sea creatures are cold-blooded. Their body temperature matches the water, so Bergmann's rule shouldn't apply, right? Except it does, for a different reason. Cold water holds more dissolved oxygen than warm water, and according to a 1999 study published in Biological Bulletin, oxygen availability may directly influence maximum body size in deep-sea organisms. The study found that in amphipods, small crustaceans, maximum size correlated strongly with dissolved oxygen levels. Species living in cold, oxygen-rich, deep water grew significantly larger than species in warmer, oxygen-depleted zones. The reason? Larger bodies require more oxygen to sustain, but they also have more efficient oxygen delivery systems. In oxygen-rich environments, the benefits of large size, metabolic efficiency, lower predation risk, outweigh the costs, higher absolute oxygen demand. In oxygen-poor environments, the calculus reverses. Cold deep sea water, saturated with oxygen, removes one of the main constraints on size, and evolution responds by pushing body size upward. But predation also plays a role. Predation pressure in the deep sea is dramatically lower than in shallow water. According to studies on deep-sea brachiopods, predation rates at abyssal depths are nearly an order of magnitude lower than in coastal ecosystems. Why? Fewer predators, lower population densities, slower movement speeds, less frequent encounters. In shallow water, being small helps you hide, being fast helps you escape. But in the deep sea, where encounters are rare and predators are often ambush hunters, rather than pursuit predators, size becomes a defense. A giant isopod is too large for most deep-sea predators to handle. Its thick exoskeleton is difficult to penetrate. Even if attacked, its size means it can survive injuries that would kill smaller animals. So reduced predation pressure removes another constraint on size, allowing evolution to favour gigantism for metabolic reasons without suffering predation costs. Put it all together. Buoyancy enables large size. Kleiber's law makes it metabolically advantageous. Cold temperatures increase oxygen availability. Reduced predation pressure eliminates one of the main costs. Food scarcity makes efficiency paramount. The result? Gigantism becomes the dominant strategy, but it's not universal. Deep-sea gigantism applies to specific groups: crustaceans, cephalopods, cnidarians, some eels. But not all deep-sea creatures are giants. In fact, the smallest organisms in the deep sea, meofauna, creatures smaller than 1 millimeter, show the opposite trend. They get smaller with depth, not larger. Why the difference? It comes down to lifestyle and feeding strategy. Gigantism works for scavengers and predators, organisms that need to travel, search, and consume large prey items when found. For them, metabolic efficiency and the ability to fast for long periods are critical. But for filter feeders, detritus feeders, and animals that glean organic particles from sediment, small size is advantageous. Smaller bodies have higher surface area to volume ratios, which makes passive feeding more efficient. They can extract more nutrients per unit of body mass from the thin film of organic material coating surfaces. So the deep sea doesn't impose one single size solution. It creates selective pressures that favour different sizes depending on how you make a living. But for the giants, the creatures that capture our imagination, the common thread is patience. Deep-sea giants move slowly. The giant squid, despite its size, is thought to be a relatively sluggish swimmer. The giant isopod crawls across the seafloor at a glacial pace. Greenland sharks, which can grow to 20 feet and live over 400 years, swim at less than a mile per hour. They're among the slowest moving sharks in the ocean. This isn't weakness. It's strategic. Movement requires energy. In an environment where energy is precious, you conserve it. You wait. You ambush. You scavenge. You do everything as slowly as possible, expending the minimum necessary to survive, and gigantism supports this lifestyle. Larger bodies store more energy. They can afford to wait longer. They can survive the lean times between windfalls. But there's a cost. Giant deep-sea creatures reproduce slowly. They grow slowly. They reach sexual maturity late. Their populations are small, their numbers scattered across vast areas. This makes them vulnerable. When deep-sea fishing or mining disrupts their habitat, recovery takes decades or centuries. A giant isopod that can live 5 years without food might take 10 years to reach maturity. If you remove a significant portion of the population, replacement rates are agonizingly slow. The giants of the deep have traded speed for endurance, rapid reproduction for longevity. They've optimized for a stable, unchanging environment where patience is rewarded and haste is wasteful. But that strategy assumes the environment stays stable, and increasingly human activity is disrupting the deep sea. Trawling, mining, pollution, climate change, altering temperature and oxygen levels. The giants evolved over millions of years to thrive in stillness. Now that stillness is breaking, and the same adaptations that made them successful Slow metabolism, low reproductive rates, extreme specialization make them fragile when conditions change quickly. The deep ocean taught us that life can grow enormous when freed from gravity and optimized for scarcity. But it's also teaching us that giants, despite their size, can be surprisingly fragile. Which brings us to the final paradox of deep-sea life. How do you reproduce when finding a mate is statistically nearly impossible, when individuals are scattered across millions of cubic miles, when your entire species might number in the hundreds or thousands across an entire ocean basin? The answer involves some of the strangest reproductive strategies evolution has ever produced. In 1925, a British ichthyologist named Charles Tate Regan was examining a female anglerfish specimen when he noticed something odd. There were small, parasitic-looking growths attached to her belly. He assumed they were parasites, perhaps some kind of deep-sea barnacle or worm. They weren't. They were males, fused males that had bitten into the female's tissue, released an enzyme that dissolved the boundary between their bodies, and permanently attached themselves. Their circulatory systems had merged. Their tissues had fused. They'd given up independence, given up feeding, given up almost everything except their reproductive organs. They'd become living sperm banks, carried through the abyss by females who would fertilize eggs on demand. This is sexual parasitism, and it represents one of the most extreme solutions to the fundamental problem facing all deep-sea life. In a habitat so vast and so empty, how do you ever find a mate? Consider the statistics. The deep ocean is the largest habitat on Earth, roughly 1.3 billion cubic kilometers of water. Most deep-sea species have small population sizes, often numbering in the thousands or tens of thousands, spread across entire ocean basins. Individual organisms can go years without encountering another member of their species. For animals that reproduce sexually, this creates an existential crisis. Sexual reproduction requires two individuals to find each other in the right place, at the right time, when both are reproductively mature. In a coral reef, that's challenging but manageable. In the abyss, it's statistically nearly impossible. Evolution has responded with strategies so bizarre they seem like science fiction. Let's start with those anglerfish males. In many deep-sea anglerfish species, particularly those in the suborder Ceratidae, males are dramatically smaller than females, sometimes 1/10 the size. They have enormous eyes relative to their body, which makes sense. Their entire purpose is finding a female in the dark. Once a male hatches, he has a limited window. He doesn't feed. He can't. His digestive system is vestigial, sometimes entirely absent. His olfactory organs, however, are massively developed. He can detect pheromones, chemical signals released by females, across vast distances, following concentration gradients through the water like a bloodhound following a scent trail. When he finally finds a female, he bites into her skin, usually near the belly or flanks. His mouth releases enzymes that break down tissue boundaries. Within days, the circulatory systems merge. His blood vessels connect to hers. Nutrients from her bloodstream keep him alive. His body degenerates. Eyes shrink, fins atrophy, internal organs disappear. What remains is essentially a pair of gonads with a blood supply, permanently grafted onto the female. When she's ready to spawn, hormones in her bloodstream trigger sperm release. She controls the timing. He's along for the ride. Some female anglerfish carry multiple males, sometimes 3 or 4 fused individuals scattered across her body. She's created her own travelling harem, ensuring she'll never need to search for a mate again. It's parasitism. It's also partnership. Neither sex could reproduce alone in the abyss. The male gets transportation, nutrition, and guaranteed mating opportunities. The female gets on-demand fertilization without the nearly impossible task of finding a mate when she needs one. It's disturbing from a human perspective. From an evolutionary perspective, it's brilliant. But sexual parasitism is only one solution. Other species use different strategies, equally extreme. Take synchronous spawning. In shallow water coral reefs, we see mass spawning events where hundreds of species release eggs and sperm simultaneously on the same night, usually coordinated with lunar cycles. It's spectacular, turning the water into a blizzard of gametes. The deep sea has no moon, No visible lunar cycle, no seasons in the traditional sense. Time in the abyss is almost meaningless. Yet some deep-sea species still manage to synchronize reproduction. Research published in Deep Sea Research in 2019 by biologist Mance Preed and colleagues found evidence of synchronized spawning in deep-sea fish populations based on gonad development patterns across specimens collected at similar times. How do they coordinate without environmental cues? Pheromones again. Chemical signals that don't just attract mates but can trigger reproductive readiness in other individuals who detect them. One fish reaches sexual maturity and begins releasing spawning pheromones. Others detect these chemicals and their own reproductive systems respond, accelerating egg or sperm development. The effect cascades through the local population. Within a few weeks, most mature individuals in the area are ready to spawn simultaneously. When spawning begins, the release of massive amounts of pheromones triggers a chain reaction. Everyone spawns at once, maximizing the chances that eggs and sperm find each other in the water column. It's chemical coordination, a form of distributed decision-making. No single individual controls the timing. The population decides collectively through molecular communication. Then there's the strategy of producing enormous numbers of offspring and hoping a few survive. Deep-sea fish like the ocean sunfish, mola mola, can produce up to 300 million eggs in a single spawning event. 300 million. It's the highest fecundity of any vertebrate on Earth. Why so many? Because mortality is astronomical. Deep-sea larvae are tiny, fragile, and drift in the water column for weeks or months. They're eaten by everything. Predation rates exceed 99.9%. Of those 300 million eggs, maybe a few dozen will survive to adulthood. It's the opposite strategy from mammals or birds, which invest heavily in a small number of offspring and provide parental care. Deep-sea species go for volume, broadcast spawning, release millions of gametes into the water, let currents mix them, and hope random chance brings enough together to maintain the population. There's no care, no protection. The adults spawn and disappear. The offspring are on their own from the moment of fertilization. This works only because deep-sea adults are long-lived. If a fish lives 80-100 years and spawns multiple times, producing millions of eggs each time, the cumulative odds shift in favor of successful reproduction. But it's a brutal strategy. Most individuals will never reproduce. Most lineages die out. Only a tiny fraction succeed. Compare this to deep-sea octopuses, which take the opposite approach. Female deep-sea octopuses in the genus Granellodon, particularly Granellodon boreopacifica, have been observed guarding their eggs for over 4 years. 4 years. That's the longest egg-brooding period of any known animal. In 2014, researchers from the Monterey Bay Aquarium Research Institute published observations of a single female G. boreopacifica at 4,600 feet depth, who remained in the exact same spot for 53 months, never leaving her clutch of roughly 160 eggs. She didn't feed. She constantly circulated water over the eggs with her arms, keeping them clean and oxygenated. By the end, she was emaciated, skin pale and torn, barely moving. When the eggs finally hatched, she died. She'd invested everything, literally her entire body's energy reserves, into ensuring those 160 offspring had the best possible start. No feeding, no movement, no recovery, just protection, and then death. Why such extreme maternal investment? Because in the deep sea, larvae survival rates are abysmal. Surface-dwelling octopuses produce tens of thousands of eggs and provide minimal care. Their larvae are planktonic, drifting with currents, feeding themselves. Most die, but enough survive because conditions are relatively rich. Deep-sea octopuses can't afford that strategy. Food is too scarce. Larval development takes too long. If you release planktonic larvae into the abyss, they'll starve before reaching independence. So deep-sea octopuses produce fewer, larger eggs with more yolk. Each one is a survival package, a miniature food supply. And the mother guards them until they're developed enough to hunt on their own. It's the only way to give them a fighting chance. She dies so they can live. There's something almost unbearable about that image. A single female in the darkness, starving slowly, arms moving gently over eggs she'll never see hatch. It's devotion at the cellular level. Evolution has optimized her biochemistry to shut down everything except the imperative to protect. reproduction in the deep sea isn't about pleasure or bonding or elaborate courtship. It's logistics. It's solving an impossible equation. How do you ensure genetic continuity when individuals are scattered across millions of square miles, when energy is scarce, when meeting a mate might happen once in a lifetime or never? The solutions are as varied as the species. Some fuse permanently, some coordinate chemically, some spawn millions of offspring, some dedicate years to a handful, but they all work. They've worked for millions of years. The deep ocean is full of life, reproducing, continuing, persisting, against odds that would seem to make it impossible. And that tells us something profound about evolution. It doesn't aim for elegance or efficiency or beauty. It aims for whatever works. And if what works is males permanently grafted to females, or mothers starving to death over eggs, or fish broadcasting 300 million gametes into the void, then that's what evolution produces. Life doesn't care how strange it looks to us. It cares about continuation. Which brings us to the final piece of the puzzle, time. Because all of these adaptations, the slow metabolisms, the extreme reproductive strategies, the gigantic bodies and patient hunting, evolved over timescales we can barely comprehend. The deep ocean has been stable for hundreds of millions of years. That stability allowed specialization at a level impossible in more dynamic environments. Species could fine-tune every aspect of their biology to extract maximum advantage from an unchanging habitat. But that stability is ending, and the creatures adapted to thrive in stillness are now facing change they're not equipped to handle. For hundreds of millions of years, the deep ocean has been the most stable environment on Earth. Temperature barely fluctuates. Pressure remains constant. Seasons don't exist. Light never penetrates. The rhythms of surface life, daylight, weather, tides, fade into irrelevance at depth. This stability allowed life to specialize in ways impossible elsewhere. A giant isopod can afford a metabolism so slow it goes years without eating because its environment won't suddenly change. An anglerfish male can afford to permanently fuse to a female because resource predictability means both will survive. A deep-sea octopus can afford to starve for 4 years guarding eggs because her offspring will hatch into a world that looks exactly like the one she knew. Evolution thrives on stability. Give a lineage millions of years in unchanging conditions, and it will optimize every aspect of its biology to extract maximum advantage from that specific set of circumstances. It will shed unnecessary features. It will fine-tune metabolic pathways. It will become exquisitely, beautifully adapted. But specialization is a double-edged sword. The more finely tuned an organism becomes to a specific environment, the more vulnerable it is to change. And the deep ocean is changing, not slowly across evolutionary timescales that allow adaptation, rapidly, within decades, within years, faster than deep-sea life can respond. The changes are coming from multiple directions at once, each compounding the others, and most of them trace back to human activity. Start with temperature. Deep-sea temperatures, long stable, are now rising as climate change warms surface waters and heat slowly diffuses downward. The changes are small, fractions of a degree per decade, but for organisms adapted to extreme stability, even small changes matter. Temperature governs metabolism. Warmer water means higher metabolic rates, which means organisms need more oxygen and more food to sustain themselves. But the deep sea doesn't have more food to give. Production is already at the limit of what can sink from the surface. If metabolic demands increase while food supply remains constant or decreases, organisms starve. Some species might adapt by moving deeper, following their preferred temperature range as isotherms shift. But many deep-sea species are already near the limits of pressure tolerance. There's nowhere colder to go. The abyss ends at the seafloor. Then there's oxygen. The ocean is losing oxygen. As surface temperatures rise, water holds less dissolved gas. Warm water is less dense, which strengthens stratification, the layering of the ocean into distinct temperature zones that don't mix well. When stratification increases, less oxygen-rich surface water circulates to depth. Oxygen minimum zones are expanding both horizontally and vertically, pushing into regions that were previously well oxygenated. For species already living at the edge of hypoxia, this is catastrophic. There's no adaptation fast enough to compensate for suffocation. Animals that spent millions of years evolving to tolerate low oxygen suddenly find themselves in conditions where even their extreme adaptations aren't enough. Then there's acidification. The ocean absorbs about a quarter of the carbon dioxide humans emit into the atmosphere. When CO2 dissolves in seawater, it forms carbonic acid, lowering pH. The surface ocean has already acidified by about 30% since the Industrial Revolution began. That acidification is now spreading to depth. Many deep-sea organisms have shells or skeletons made of calcium carbonate. Corals, mollusks, echinoderms, certain plankton. In acidic water, calcium carbonate dissolves, not quickly, but inevitably. The chemistry shifts against them. Building and maintaining shells becomes harder. Energy that should go toward growth or reproduction gets diverted to structural maintenance. Some deep-sea species might not survive at all, as pH drops below thresholds their biochemistry can tolerate. These are the indirect impacts, the creeping changes in baseline conditions that alter the fundamental habitability of the deep ocean. They're slow, hard to detect, and nearly impossible to reverse once set in motion. But there are also direct impacts, physical destruction, and these are happening now at scales that dwarf anything in the deep ocean's recent history. Bottom trawling. Bottom trawling is a fishing method where weighted nets, some with metal doors weighing several tons and foot ropes studded with steel balls, are dragged across the seafloor to catch fish. The nets scrape, crush and bulldoze everything in their path. On soft sediment bottoms, mud and sand, trawling kills in fauna, resuspends sediment and disrupts feeding and reproductive behaviours. Studies in the Mediterranean found that chronically trawled areas showed organic matter content reduced by up to 52%, meiofauna abundance down 80%, and biodiversity reduced by 50%. But the worst damage occurs on hard substrates, seamounts, ridges, cold water coral reefs. These are biodiversity hotspots, the rainforests of the deep. Deep-sea corals can be hundreds to thousands of years old, with some dated at over 4,000 years. They grow millimeters per year, building massive reef structures that provide habitat for thousands of species. A single trawl pass destroys them in seconds. Research on seamounts off Tasmania found that even 14 years after trawling stopped, there were no signs of recovery. The community structure remained degraded, resembling actively trawled sites. 14 years for ecosystems that took millennia to develop. The United Nations World Ocean Assessment found that bottom trawling has caused widespread, long-term destruction globally, and that recovery may take centuries to millennia. We are turning portions of the deep sea floor into deserts. Not metaphorically, literally. Areas that once supported diverse communities are now barren, dominated by a handful of opportunistic species adapted to disturbance. And trawling isn't slowing down. As coastal fisheries collapse from overfishing, industrial fleets push deeper, targeting slow-growing species like orange ruffy, which can live over 200 years and take decades to reach reproductive maturity. These fish evolved life histories optimized for stability, slow growth, late maturity, long lifespans, strategies that work when mortality is low and predictable, but they can't withstand industrial harvest. Populations crash within years of exploitation beginning, and recovery, if it happens at all, takes generations. We're mining fish, not fishing sustainably, extracting accumulated biomass and leaving behind degraded ecosystems. But if bottom trawling is bad, what's being proposed next is potentially catastrophic. Deep sea mining. The seafloor holds vast mineral deposits, polymetallic nodules rich in cobalt, nickel, copper, and rare earth elements. These nodules formed over millions of years as metals precipitated from seawater and accumulated layer by layer on the ocean floor. They grow at rates of millimeters per million years. They also serve as habitat. The rough surfaces provide attachment sites for sponges, corals, anemones. The nodules create structural complexity in otherwise flat abyssal plains. Studies show that the majority of biodiversity in nodule fields lives directly on the nodules themselves. Deep-sea mining would vacuum these nodules up, along with everything attached to them, along with the top few inches of sediment. The mining vehicles would churn the seafloor, creating massive sediment plumes that drift for kilometers, smothering organisms downwind. the wastewater from mineral processing, a toxic slurry of sediment, crushed animals and chemicals, would be dumped back into the ocean, creating discharge plumes that spread over vast areas. Mining would generate intense noise and light pollution in environments that are naturally silent and dark. Sound and light could disrupt communication, reproduction, and feeding behaviours in ways we can't yet predict and recovery. A study published in Nature in 2025 examined a site where experimental nodule collection occurred 40 years earlier. 4 decades later, biological communities had not recovered. Many species were still absent. The seafloor showed persistent physical changes 40 years and no recovery. For an ecosystem that evolved over tens of millions of years, scientists warn that biodiversity loss from deep-sea mining would be inevitable and most likely irreversible. Species could go extinct before we even discover they exist. Ecosystem services, carbon sequestration, nutrient cycling, fisheries support could collapse. Despite these warnings, the International Seabed Authority has been negotiating regulations to allow commercial mining to begin, potentially by 2025. Companies are already testing equipment. Norway announced in 2023 that it would proceed with exploration of deep-sea mining in its waters, becoming one of the first nations to authorize such activity. The rush is driven by demand for batteries, electric vehicles, renewable energy storage, electronics. Proponents argue that deep-sea mining avoids the environmental and human rights problems of terrestrial mining. No deforestation, no displaced communities, no child labour in cobalt mines. But economic analyses suggest deep-sea mining would generate minimal benefits while imposing massive environmental costs. A study by Planet Tracker estimated that deep-sea mining could destroy over $500 billion in natural capital and corporate value. Small island nations that depend on tuna fisheries could see catches drop by 10% or more if mining disrupts marine ecosystems in targeted regions. The people with the least responsibility for climate change would bear disproportionate harm from a supposed solution to it. And there are alternatives: recycling, material substitution, more efficient use of existing resources, circular economy models that eliminate the need for virgin mineral extraction entirely. Several countries, Chile, Palau, Fiji, Vanuatu, France, have called for moratoriums on deep-sea mining until comprehensive environmental assessments can be completed. They recognise what should be obvious: we don't know enough to do this safely, and once the damage is done, it can't be undone. But the pressure continues. The International Seabed Authority moves forward with regulations, test equipment is deployed, the clock is ticking, and the deep ocean, which has no voice, no constituency, no political power, faces industrialization before we've even finished exploring it. We've mapped more of Mars than we have of our own seafloor. We've named more craters on the moon than seamounts in the deep ocean. And we're preparing to mine ecosystems we don't understand, destroying habitats we've barely seen, erasing species we haven't discovered. The irony is suffocating. We look to the stars for alien life while ignoring the aliens in our own ocean. We spend billions searching for microbes on Mars while bulldozing coral reefs that took millennia to grow. We fantasize about terraforming other planets while rendering large portions of our own uninhabitable. The deep ocean survived 5 mass extinctions. It persisted through ice ages, through volcanic cataclysms, through asteroid impacts. It adapted to every change the planet threw at it across 3.5 billion years. But it never faced anything like this. It never faced a single species capable of altering global chemistry, warming the entire ocean, and physically destroying habitat at industrial scales within decades. The organisms of the deep sea evolved patience. They evolved to wait, to endure, to outlast. Their entire biology is built on the assumption that stability will continue, that time stretches on, that tomorrow looks like yesterday. That assumption is breaking. And when an ecosystem optimized for stability faces rapid change, the result isn't adaptation, it's collapse. The giant isopods, the anglerfish, the deep-sea octopuses, the coral forests, they're not equipped for this. Their slow metabolisms can't speed up. Their low reproductive rates can't increase. Their specialized biochemistry can't pivot. Evolution doesn't work that way. Adaptation requires variation, selection, and time. Deep-sea species have variation, though often less than shallow water relatives due to stable conditions and small population sizes. They're certainly under selection pressure now, but they don't have time. Recovery from disturbance, when it happens at all, takes decades to centuries. Extinction happens in years. We're forcing deep-sea life to play a game they can't win, under rules evolution never prepared them for, on timescales that make adaptation impossible. And the tragedy is, it's not even necessary. We don't need to mine the deep sea. We don't need to trawl every seamount. We could choose differently. We have the technology, the knowledge, and the resources to meet human needs without destroying the last great wilderness on Earth. But choice requires will, and will requires caring, and it's hard to care about what you can't see. The deep ocean is out of sight. Its inhabitants are strange, unfamiliar, sometimes unsettling. They don't have big eyes or fur. They don't trigger mammalian empathy circuits. A giant isopod doesn't inspire the same protective instinct as a panda. But they're no less alive, no less the product of billions of years of evolution, no less worthy of existence, and they might be teaching us something we desperately need to learn: that life doesn't require familiarity to be valuable, that intelligence doesn't need brains to be real, that Earth contains worlds as alien as any we'll find in space. The ocean didn't evolve life, it invented another one. A parallel experiment in what biology can become when freed from terrestrial constraints, when pressure replaces gravity, when darkness becomes the canvas, and chemistry becomes language. For 4 billion years, life found ways to persist. In boiling vents and frozen plains, in acid and vacuum and crushing dark. It adapted, diversified, filled every niche, turned every challenge into opportunity. The deep ocean represents the final chapter of that story on Earth. The last frontier where evolution still writes new possibilities, where undiscovered species outnumber known ones, where the fundamental rules of life are being tested in ways we're only beginning to understand. And we're about to close the book, not through malice, through indifference, through the banal logic of extraction that sees resources instead of ecosystems, commodities instead of communities, profit instead of irreplaceable complexity. The deep ocean can't speak for itself. Its inhabitants can't protest, can't vote, can't appeal to our reason or compassion. They exist in silence, in darkness, beyond our sensory world. Which means the responsibility falls to us, to recognize what we're losing before it's gone, to understand that alien doesn't mean expendable, to accept that some places should remain beyond the reach of industry, not because they lack value, but because their value transcends extraction. The creatures of the deep sea evolved every strategy imaginable to survive in an impossible place. They solved problems physics said couldn't be solved. They built communities in emptiness, found food in starvation, reproduced against impossible odds. They survived everything Earth could throw at them, everything except us. We began this journey asking how life could be so different beneath the waves, how evolution could produce organisms so alien, so strange, so unlike anything on land. The answer is simple. The ocean isn't like land. It never was. It's a three-dimensional volume where gravity weakens, where pressure dominates, where light dies and chemistry speaks. It's a physical regime so different from the terrestrial surface that life had to reinvent itself to exist there. And in that reinvention, it created wonders. Bioluminescence that turns darkness into language. Pressure-adapted proteins that function at depths that would crush steel. Metabolisms so efficient they can extract energy from near nothing. Reproductive strategies that solve the mathematics of loneliness in the largest habitat on Earth. These adaptations aren't just curiosities. They're proof that life is more flexible, more creative, more resilient than we imagined. That the rules we think govern biology are just the rules governing terrestrial biology. That the universe of possible life forms is vastly larger than the tiny subset we see on land. The deep ocean shows us what life becomes when the constraints change, and in doing so, it hints at what we might find elsewhere. On Europa's ice-covered ocean, on Enceladus, on worlds we haven't discovered yet. But first, we have to protect the alien world we already have. Because if we can't preserve the biodiversity on our own planet, in our own ocean, we have no business reaching for others. The deep sea is still here, still alive, still evolving. The The giant isopods still scavenge, the anglerfish still hunt, the octopuses still guard their eggs in the dark, but the window is closing, the stability is ending, and the creatures who evolved to endure are running out of time to adapt. The choice is ours. We can be the generation that industrialized the abyss and erased ecosystems before we understood them, or we can be the generation that recognized what we were about to lose and chose differently. We can see the deep ocean as alien, yes, but also as kin, as fellow travelers through deep time, as proof that Earth is stranger and richer than any planet we'll visit in our lifetimes. The ocean didn't just evolve life, it evolved possibilities, and every species we lose is a possibility that will never exist again. Every ecosystem we destroy is a question we'll never get to ask. We started by marveling at how different life is beneath the waves. We end by realizing that different doesn't mean separate, that the deep ocean, for all its strangeness, is still Earth, still our biosphere, still part of the planetary web that sustains all life, including ours. And if we're willing to destroy it simply because we can't see it, what does that say about how we'll treat everything else invisible to us? Future generations, other species, the thin envelope of atmosphere and ocean that makes this rock habitable? The deep sea asks nothing of us except what all life asks: room to exist, time to continue. The dignity of not being erased by indifference. It's a small request, and it's the least we owe the creatures who proved that Earth is more alien, more beautiful, and more full of possibility than we ever imagined. They've been evolving in the dark for hundreds of millions of years. The least we can do is let them continue. We live on a planet we barely know, a world of light and land, of wind and stone, but beneath it all lies another Earth. It moves to its own slow rhythm. It breathes with unseen weather. It remembers everything. We call it the ocean, but that word is too small for what it truly is. Because this is not just water, it's the living bloodstream of the planet, a vast fluid world that shaped every breath, every storm, every heartbeat in the history of life. From space, it looks like infinity, a single body of blue, hiding the greatest wilderness we've never seen. Before there was land, there was water. Before there was life, there was the sea. It created us, it sustains us, and it will outlive us. This is the ocean, Earth's last frontier. More than 70% of our world is covered by it. It holds 97% of all water on Earth and drives nearly every process that sustains life above the waves. The rain that fills our rivers, the air we breathe, the clouds that cradle continents, all begin with the sea. We often call space our final frontier, yet the greatest unexplored realm is the one surrounding us. We've mapped the surface of Mars in higher resolution than our own ocean floor. We've sent probes beyond the edge of the solar system, but still, less than 5% of Earth's seabed has been seen, let alone understood. Every sonar scan, every descent, every sample taken from the depths reveals something that challenges what we thought we knew about our home. The irony is profound. In our search for other worlds, we've overlooked the alien world beneath our feet. To the earliest explorers, the ocean was infinite, a mystery stretching past the horizon. Its depth was a myth, its creatures, superstition, its power, divine. Today, science has replaced myth with measurement, but even now the sea resists definition. It's too vast, too dynamic, too alive to be reduced to data points on a map. The ocean is not one uniform body. It is a collection of systems layered and intertwined, a living machine made of currents, temperature, chemistry, and life. And within that complexity there is balance, a constant negotiation between heat and cold, carbon and oxygen, light and darkness. It is both cradle and climate engine, both destroyer and saviour. The ocean shapes everything it touches. Over millions of years it sculpted the continents, ground down mountains, and carried their dust to form new land elsewhere. It wrote the coastlines that define our geography and built the atmosphere that allows us to breathe. Even now, the ocean regulates the global thermostat. It absorbs and releases heat, controls rainfall and drought, and determines the strength of storms. Every breath of wind that crosses a city, every drop of rain that falls on a forest, begins as an idea whispered by the sea. And yet, for all its power, the ocean remains strangely invisible. We live alongside it, travel over it, extract from it, and rely on it completely, but we rarely see it as a world in its own right. The surface is all most of us ever know, a glittering skin of reflected sky, a shifting mirror of weather and light. But beneath that thin boundary lies a place as foreign to human experience as the surface of another planet. A single drop of seawater can contain more than 1 million living organisms. Each one, plankton, bacteria, larvae, is a tiny engine in a vast planetary network, recycling nutrients and capturing sunlight to feed the world above. Collectively, these microscopic drifters produce over half the oxygen in our atmosphere. Every second breath you take came from the ocean. We tend to think of the ocean as empty, but it's the most biologically active region on the planet. From its surface to its deepest trench, it sustains forms of life so diverse and so specialized that entire ecosystems can exist within a single cubic meter. To understand Earth is to understand that the ocean is the planet. The continents are interruptions islands in a single interconnected sea. Our species has spent centuries trying to chart this water world. The first oceanographers dragged weighted lines from sailing ships to measure depth. The Challenger expedition of the 1870s crossed the globe cataloguing species no one had imagined. Since then, submersibles, satellites and sonar arrays have deepened our perspective, but not our mastery. The deeper we look, the more the ocean confounds us. At the surface, sunlight drives motion. Wind pushes waves that transfer energy across thousands of kilometers. Beneath them, temperature and salt govern density, the forces that steer the great currents linking every sea. These invisible rivers of water shape climate and life everywhere they flow. They connect the Arctic to the tropics, the Atlantic to the Pacific, and the atmosphere to the land. The ocean, in this sense, is not just part of the planet, it is the planet's circulation, its bloodstream. But there's another kind of current that flows through the sea: time. Every layer of sediment, every trapped bubble of gas, every chemical trace preserved in the deep records the history of the world. In the ocean's depths, time moves differently. The water itself can be centuries old, formed at the poles, sinking into the abyss, and only now returning to the surface. To study the sea is to read the biography of Earth, written in waves instead of words. Perhaps that's why the ocean still feels so mysterious. It's not simply that it's unexplored, it's that it operates on scales beyond human intuition. We measure time in years and generations. The ocean measures it in cycles and epochs. We live by sight and air. The ocean exists by pressure and flow. It is not hostile, only indifferent. And in that indifference lies a kind of purity, a reminder that Earth is more than habitat. It's an organism, vast and alive, and we are part of its respiration. The story of the ocean is not one of discovery, but of perspective. Each expedition, each dive, each new dataset pulls back a veil, revealing that the ocean is not just the background of life, but its foundation. Everything we know, everything we depend on began here, and everything we do still returns here. This is where the story begins, not with what we build upon the land, but with the water that built the land itself. Because to understand the future of Earth, we must first descend into the world we've barely begun to see. The first thing we see when we look at Earth from space isn't land or clouds or cities. It's blue, a colour so deep and dominant that it erases everything else. Our planet is not a world of continents surrounded by ocean, it's a world of ocean decorated with continents. From orbit, the ocean is alive with motion. Swirls of turquoise mark coral reefs and continental shelves. Dark gyres twist across the mid-latitudes like slow-turning gears, and along the equator, a shimmering belt of cloudless calm marks the place where the planet breathes out. What looks like stillness from above is, in truth, constant movement, the circulation of heat, water, and life. Every gust of wind across the surface transfers energy into the sea, sculpting waves that roll across thousands of kilometers. Some will travel halfway around the planet before breaking against a shore. Others will merge and collapse, disappearing back into the body of the ocean that made them. Each wave is born, lives, and dies in a matter of seconds, yet collectively, they shape the face of every coastline on Earth. The ocean's surface is only a few millimeters thick, but it is the boundary that connects everything below to everything above. It's where sunlight meets seawater and where invisible gases pass in and out of the atmosphere. Carbon dioxide dissolves here, oxygen escapes here. This thin film regulates the chemistry of the air itself. In every sense, it is the skin of the planet. Beneath that skin, light penetrates only about 200 meters, a fragile layer scientists call the photic zone. Within this narrow band of brightness, life explodes in diversity and abundance. Tiny drifting plants known as phytoplankton capture sunlight through photosynthesis, using it to convert carbon dioxide into oxygen and sugar. They are so numerous that they can be seen from space, colouring entire ocean basins in swirls of green and gold. These microscopic organisms form the foundation of the marine food web, feeding everything from krill to whales. Collectively, they generate more than half the oxygen in Earth's atmosphere. Every second breath you take is thanks to them. But the ocean surface is not just a biological wonder, it's a physical one. Sunlight warms the upper layers unevenly, creating gradients in temperature and density. Where warm and cool waters meet, currents form. These currents act like conveyor belts, moving heat across the planet. Warm water from the tropics drifts toward the poles, while cold water from the poles sinks and returns along the seabed. This global circulation is Earth's climate engine, distributing energy with quiet precision. The ocean doesn't just absorb heat, it moderates it. Because water holds vast amounts of thermal energy, it buffers the planet from extremes. The difference between a frozen world and a habitable one is measured in the ocean's capacity to store warmth. When the sea warms, the air follows. When the sea cools, the winds and clouds respond. Every storm, every drought, every gentle breeze begins as a whisper between ocean and sky. The living surface is also the world's meeting place. Here, life from the deep mingles with life from the air. Flying fish leap from waves to escape predators. Seabirds dive like arrows into the glittering schools below. Whales breach through the boundary, pulling the unseen ocean into view for just a moment before vanishing again. This is where the lines between worlds blur, where the ocean reminds us that air and water are just different states of the same cycle. Yet this delicate equilibrium is constantly tested. Wind, temperature, and salinity form a dynamic balance that can shift with even the smallest change. When polar ice melts, it dilutes surface salinity, altering the density of seawater and slowing major currents. When surface waters warm, they expand, raising sea levels. When the air absorbs more carbon dioxide, the ocean follows, forming carbonic acid that dissolves coral reefs and shellfish. The skin of the planet is sensitive, and it responds to our every action. Despite this fragility, the ocean is resilient. Its systems are self-correcting on timescales much longer than our own. A single current may take 1,000 years to complete its loop around the world. To the ocean, human centuries are brief fluctuations, but even resilience has limits. When pressure is applied faster than the system can respond, change accelerates, and equilibrium becomes something new. It's easy to think of the ocean's surface as familiar. We sail it, swim in it, look out over it from our cities and coasts. But this is not the ocean we truly live with, it's only the ocean we can see. Beneath the waves lies another realm entirely, where light becomes memory and colour fades to blue, then to black. It's a descent not just into water, but into time. Pressure increases, temperature drops, and the physics of survival shift into new territory. For most of human history, we thought of that deep world as lifeless, a cold abyss too hostile for biology. But we were wrong. The truth is far more extraordinary. In the depths below the living surface, life has adapted to the impossible. There in the darkness it glows. It stretches, compresses, and bends the very rules of existence. To enter the deep ocean is to enter a place that redefines what it means to be alive. And that is where we go next, down into the twilight, through the silence, into the heart of the unknown. To descend into the sea is to leave behind the world as we know it. Every metre downward feels like moving through time backward, away from the familiar, into the deep history of the planet. Light fades quickly in seawater. Within just a few dozen metres, red disappears, then orange, then yellow, until only blue remains. Below 200 metres, even that vanishes. Here begins the ocean's twilight zone, the Mesopelagica realm between day and night, where sunlight flickers but never truly arrives. The pressure rises with each descent 1 atmosphere for every 10 metres. At 1,000 metres, it's 100 times greater than at the surface. Metal creaks, air compresses, even sound changes character, moving faster, sharper, like a vibration through glass. In this perpetual dusk, light becomes currency. Creatures that live here have evolved to make their own. Tiny lanternfish drift in vast shoals, each dotted with photophores, organs that glow like stars. Siphonophores, jelly-like colonies stretching dozens of metres, shimmer in pulses of green and blue. Bioluminescence is communication, camouflage, and lure the language of the deep. A single flash can mean danger. A faint glow along the belly of a fish can erase its shadow, making it vanish against the faint light above. To human eyes, this world feels alien, but to the species that inhabit it, it is the centre of existence. Life here follows an ancient rhythm. Every night, when surface waters cool and darkness falls, billions of creatures migrate upward from the twilight zone to feed. They rise silently, forming a vast invisible tide of life. Just before dawn, they sink again, carrying carbon and nutrients into the deep ocean. It is the largest migration on Earth, a movement so immense it influences the global carbon cycle. And it happens every single night. As we descend further, light fades entirely. This is the midnight zone, the bathed pelagica realm of total darkness, stretching from 1 kilometre down to 4. No sunlight has ever touched this place. Here, pressure is immense, temperatures hover near freezing and food is scarce, yet even here life endures. The animals of the midnight zone are built from compromise, their bodies soft, their metabolism slow, their senses sharpened in ways we can barely imagine. Some, like the anglerfish, carry lures of living light, dangling bioluminescent bait before jaws lined with transparent teeth. Others, like the vampire squid, drift with outstretched arms covered in photophores, creating a living constellation of defense. There are species that have no pigment, no eyes, and others whose eyes are enormous, designed to capture even the faintest glimmer from above. In this darkness, survival depends on efficiency. Energy is precious, movements are slow, encounters are rare. Predators and prey exist in equilibrium, each adapting to the other in an endless evolutionary dialogue. Occasionally we find reminders that the surface still matters here. Marine snow, a gentle rain of detritus, plankton and decaying matter, drifts endlessly downward. It is the deep ocean's lifeline, feeding creatures that will never see sunlight. When that snow reaches the seafloor, it accumulates into layers that hold the memory of ages' chemical archives of ancient climates, volcanic eruptions, even mass extinctions. The ocean's floor is not dead ground. It's a slow-moving library, preserving everything the world forgets. At around 4,000 metres, the bottom appears a vast plain stretching farther than any desert. This is the abyssal zone. The temperature never rises above 2 degrees Celsius. There are no seasons, no day or night. Only the slow drift of silt and the faint vibration of distant tectonic motion. And yet, life continues. Sea cucumbers inch across the sediment, filtering nourishment from the dust. Brittle stars extend their arms to catch invisible currents. Even here, miles beneath storms and sunlight, the biological pulse of Earth continues to beat. Occasionally the monotony of the abyss is broken by vents, fissures where seawater meets molten rock. Superheated fluids surge upward, carrying metals and chemicals that crystallize into towers and chimneys. Around these vents, life flourishes in total darkness, drawing energy not from the sun but from the Earth itself. Here, bacteria harness hydrogen sulfide, fueling entire ecosystems of worms, crabs and clams. When scientists first discovered these vents in 1977, they realized they had found something profound: life powered by geology. A reminder that biology and planet are not separate systems, but one continuous cycle of energy and adaptation. Below even this, where the ocean trenches cut into the crust, lies the Hadal Zone, the true abyss, named for Hades, the underworld. In these trenches, the water column can reach 11 kilometers deep. The pressure is so extreme that most materials would collapse instantly. For decades, scientists believed life couldn't exist there. Then cameras were sent down and moving shapes appeared on the screen. Translucent snailfish, amphipods and shrimp-like creatures drifted calmly among the rocks, perfectly adapted to their impossible world. They don't see the ocean as hostile or alien. To them, it's normal, the only home they've ever known. Every descent we make into these depths forces us to reimagine what life can be. Creatures here metabolize differently, reproduce differently, and age at different rates. Their existence challenges the limits of physics and biology, proof that life is not fragile but relentless. Even in this silence, the ocean is moving. Currents creep through the trenches, whispering across continents. Sediment slides like slow avalanches. Earthquakes send vibrations echoing through the water. The deep is not still, it breathes, just in a slower rhythm than our own. In its darkness, the ocean holds a strange kind of peace, a stillness that is not absence but balance. The creatures of the deep are not reminders of how far removed this world is from ours, but how closely tied we are to it. The oxygen in our lungs, the climate that sustains us, the stability of our planet, all are anchored in this hidden world. The deep sea is not a graveyard. It's the foundation of life on Earth, a vast, silent engine that has turned without pause for billions of years. And even now it continues to turn slow, unseen, essential. Every moment of every day the ocean moves, not with the chaos of waves or tides, but with the steady persistence of a planetary heartbeat. Water formed in the cold, dark polar seas sinks, sliding along the seafloor, creeping across entire ocean basins before rising again thousands of kilometers away. It's called the Global Ocean Conveyor Belt, a slow continuous circulation that connects every ocean into one single, moving system. This circulation is driven by density, by temperature and salt. Warm, fresh water is lighter and floats near the surface. Cold, salty water is heavier and sinks. Where the two meet, they mix and begin a journey that can last 1,000 years. A drop of seawater formed today in the North Atlantic might not resurface until the next millennium, emerging somewhere in the Pacific as part of the cycle's return. The scale of this system is difficult to imagine. It transfers heat around the planet, carrying warm water from the tropics toward the poles, and returning cold water back again. It is why the coasts of Western Europe remain temperate even at high latitudes, and why tropical oceans stay relatively stable despite the constant blaze of sunlight. The conveyor is the ocean's method of balance and equalizer that keeps extremes in check, but it is more than just a transport of heat. It is also the circulation of life. The upwelling zones where deep waters rise are some of the most productive ecosystems on Earth. As old water returns to the surface, it brings with it the nutrients of ages: nitrogen, phosphorus, and iron, recycled from countless generations of marine snow that fell into the depths. These nutrients fuel blooms of plankton, which feed fish, whales, and seabirds, supporting the entire oceanic food web. From the air, this cycle is invisible, yet satellites can see its fingerprint spirals of phytoplankton tracing the paths of currents, temperature gradients glowing faintly in infrared, vast eddies spinning like the slow gears of a clock. To study them is to study time itself. The energy that drives these currents is sunlight, wind, and the rotation of the Earth, all acting on the scale so immense that the human mind can only grasp them through analogy. Imagine a single breath moving through the lungs of a planet. That is the thermohaline circulation. At the surface, winds sculpt patterns we recognize. the trade winds pushing equatorial waters westward, the westerlies driving mid-latitude gyres, and the polar easterlies spinning the Arctic and Antarctic currents. Together they generate a continuous motion that binds the atmosphere and ocean into one system. Weather does not begin in the sky, it begins here, in the slow exchange of heat between air and sea. When warm surface water evaporates, it leaves salt behind, increasing the salinity of what remains and helping it sink. When polar ice forms, it rejects salt, creating dense brine that plunges downward like liquid gravity. When the ice melts, it freshens the surface, lightening it, slowing the great sinking currents. In this way, the cryosphere, the world's frozen water, acts as a throttle for the entire planetary engine. Too much melting and the engine slows, too much freezing and it accelerates. The balance is delicate, and in recent decades, we have begun to notice its tremors. The modern ocean absorbs over 90% of the excess heat trapped by greenhouse gases. This has consequences. Warm water expands, raising sea levels. Temperature gradients shift, altering the paths of major currents. In the North Atlantic, scientists are watching for signs that the deep water formation driving the conveyor may be weakening. If it slows significantly, weather patterns from monsoons to jet streams could shift. The ocean that stabilizes life could just as easily rewrite its climate. Even so, the ocean's capacity for resilience is extraordinary. Its chemistry adjusts, its circulation reorganizes, its biological systems adapt. The same water that once cycled through prehistoric seas still moves through the basins today, altered but enduring. To the ocean, our age is a moment of brief warming, a temporary imbalance in a story 4 billion years long. The engine does not stop, it changes rhythm, and within that rhythm, everything alive finds its place. Currents sculpt coastlines and carry larvae across oceans, connecting ecosystems separated by continents. They deliver nutrients that bloom into coral reefs, oxygenate the abyss, and ventilate the entire biosphere. The very sound of waves breaking on a shore is energy born thousands of kilometers away, translated through this great machinery. To understand the ocean's circulation is to understand that motion is not noise, it is memory. Each current carries information: heat from past seasons, chemistry from ancient volcanoes, sediments from forgotten rivers. It is a planetary archive in constant motion, rewriting itself endlessly but never erasing what came before. We live inside the consequences of that motion. Our agriculture, our weather, our civilizations depend on patterns set by water we will never see. The same ocean that shapes hurricanes also sustains the monsoons that feed billions. The same circulation that cools the poles warms the fields where we grow our food. We are not separate from this system, we are expressions of it. Sometimes we speak of the ocean as though it were a mechanism, a machine of physics and chemistry. But that language misses something. The ocean's engine is not mechanical, it's biological, geological and atmospheric all at once. It is life in motion, self-balancing and self-correcting, the beating heart of a living planet. And as that heart continues to pulse, it shapes not only the weather and the seasons, but the boundaries of possibility for every form of life on Earth. The same currents that connect continents also connect time. The water rising at the surface today may have been last exposed to the sky during the fall of ancient civilizations. When it rises again centuries from now, it will carry the imprint of our age, a record of our choices written into its temperature and chemistry. The ocean remembers, and its memory will shape the world yet to come. Every coastline on Earth is a collaboration between water and time. The sea never rests, and across billions of years it has carved the shape of continents, built mountains, and erased them again. It is the great sculptor of the planet's surface, and the keeper of everything that surface has ever been. The ocean is not the opposite of land, it's the reason land exists. Long before mountains rose, long before continents drifted apart, the ocean was already at work. Rain, rivers and tides eroded the early crust, grinding rock into sediment that the sea reclaimed and compressed into new layers of stone. Those layers, buried and heated, became the foundation of continents. Land was born of the ocean, and in the end it will return to it. Each grain of sand carries the memory of this cycle. Fragments of ancient rock that once formed mountains, ground down by wind and rain, carried by rivers to the coast, then rolled and polished by waves until they became part of the shore. What we call a beach is simply time made visible. Stand barefoot at the edge of the sea and you are standing on the remains of the worlds long gone. But the ocean doesn't only shape the land, it shapes the air itself. Billions of years ago, photosynthetic microbes in the sea began releasing oxygen as a byproduct of their metabolism. At first, that oxygen reacted with dissolved iron, staining the ocean rust red. Only when those iron stores were exhausted did oxygen begin to accumulate in the atmosphere. The blue sky we see today is, in a sense, an oceanic creation, the breath of an ancient sea made visible. Even now, the ocean continues to write the atmosphere's chemistry. It absorbs roughly a quarter of all carbon dioxide we emit, dissolving it into a vast reservoir that buffers the climate, but also changes the sea itself. When carbon dioxide reacts with water, it forms carbonic acid, which gradually reduces the ocean's pH. This process, ocean acidification, weakens coral skeletons and shellfish shells, subtly altering the architecture of entire ecosystems. The ocean is both absorber and victim, healer and witness. In its depths, it records these changes. Layers of sediment on the seafloor form a continuous timeline of Earth's past. Every eruption, every extinction, every shift in climate leaves a signature in those layers: volcanic ash, microfossils, isotopes of oxygen and carbon. When scientists extract cores from the seabed, they are reading a diary millions of years old. The rise and fall of temperatures, the cycles of ice and warmth, the traces of mass extinctions, all are there, preserved in grains of mud. These archives reveal a profound truth. The ocean has always been the author of renewal. Every catastrophe on land asteroid impacts, volcanic winters, global ice ages has been followed by recovery, and that recovery has always begun in the sea. When life was nearly erased, it was the ocean that sheltered what remained. From those survivors, evolution began again. The story of life is written in the tides. The earliest multicellular organisms arose in shallow marine shelves where waves and sunlight met. Fish evolved there, developing the first backbones. Eventually some left the water, breathing air and walking on land, but even in us the ocean remains. The saline composition of human blood matches that of ancient seawater. The rhythm of our hearts echoes the tides. In a biological sense, we never truly left the sea, we carried it with us. But if the ocean creates, it also destroys. It is capable of both gentle erosion and sudden violence. Storm surges, tsunamis, and rising tides reshape coasts in moments. Yet even in destruction, there is creation. Coral reefs grow from the skeletons of previous generations. Deltas form from floods, and new shorelines emerge where old ones fall away. The ocean's artistry is not permanent, it's perpetual. And just as it shapes the physical Earth, it shapes its climates. The interplay of evaporation, condensation, and rainfall forms the planet's weather systems. When warm ocean water evaporates, it feeds clouds that travel thousands of kilometers, releasing their rain over distant lands. The moisture that falls in forests, on farms, and on cities all begins in the ocean's invisible breath. Even deserts are touched by it, their winds, their seasonal changes. Their very existence depend on the ocean's control of heat and air. Without the ocean's stabilizing presence, the world would be unrecognizable. Continents would swing between extremes of heat and cold. Rainfall would vanish. Storms would rage unchecked. Life, as we know it, could not endure. The ocean does not merely support the planet, it defines it, and it holds memory far beyond any human record. Long before there were fossils or bones, the ocean was archiving time and chemistry. The ratio of oxygen isotopes in microscopic shells tells us the temperature of seas millions of years ago. The concentration of certain elements reveals the waxing and waning of ice ages. These are messages from the deep whispers from the Earth to its future inhabitants, telling them what has come before and what might come again. The ocean remembers everything. It remembers warmth and ice, life and loss. It remembers when our ancestors first looked across its horizon and wondered what lay beyond. And it will remember us. Every trace of carbon we release, every plastic fragment, every warming current, all will be recorded, layered, and sealed into the next chapter of its history. To think of the ocean as a mere body of water is to underestimate its reach. It is the planet's builder, its historian, its lungs and heart combined. It sculpts the continents, regulates the sky, writes the climate, and shelters the memory of all that has lived. It is both archive and author, both witness and creator, and through all the transformations of Earth through shifting continents, changing climates, extinctions and renewals the ocean has endured, constant, infinite, alive. It has been here since the beginning, and it will remain long after everything we know has changed. The ocean has always been our beginning, and it may yet determine our end. Every civilization that has ever risen has done so beside the sea. It carried our ships, connected our continents, and fed our cities. For millennia, it was a mystery we worshipped from the shore. Today we've measured its temperature, charted its floor, and traced its currents from orbit, but in doing so, we've begun to forget what it truly is. The ocean is not a backdrop, it is the planet itself, and we are one of its recent experiments. In a single human lifetime, we've altered its chemistry, its temperature, and its life. The ocean has absorbed over 90% of the excess heat trapped by greenhouse gases. That heat expands water, raising sea levels. It melts ice, changing salinity and slowing the great global currents that regulate climate. It disrupts winds, reshapes monsoons, and intensifies storms. The very engine that has kept our world stable for millions of years is beginning to change rhythm, and we are the cause. Carbon dioxide dissolves into seawater, forming acid that eats away at coral and shell. Rising temperatures push marine life toward the poles, disrupting food webs that evolved over millions of years. Overfishing has emptied vast regions of life. Plastic now outweighs plankton in some coastal waters. We've extracted minerals, oil, and sound, leaving scars both visible and invisible. The ocean endures, but it is not untouched. And yet, even in the face of change, it continues to give. It still moderates our weather, provides our oxygen, and stores our waste. It remains the planet's great stabilizer, even as we test its limits. No matter how advanced our technology becomes, we are still creatures of its design, products of its chemistry, governed by its climate. The more we learn about the ocean, the clearer our reflection becomes. Its currents mirror our circulatory systems. Its rhythms resemble our breath. Its capacity to adapt, to find equilibrium in chaos, mirrors our own struggles to do the same. The ocean story is not separate from ours, it is ours. We are made from its elements, sustained by its cycles, and bound to its future. When we speak of saving the ocean, we are in truth talking about saving the conditions that make our existence possible. The ocean doesn't need rescue in a sentimental sense, it will persist regardless. But whether it remains a cradle for complex life or a monument to what once was depends on us. In the deep sea, where sunlight never reaches, hydrothermal vents continue to create new ecosystems, new forms of life, new beginnings. That same creative impulse exists within us. We are capable of destruction, yes, but also of renewal. For every reef that bleaches, there are scientists cultivating corals in nurseries and replanting them. For every polluted coastline, there are communities restoring mangroves and seagrasses that pull carbon from the air and shelter fish once thought lost. The ocean's capacity for recovery mirrors our capacity for change. Both depend on time, persistence, and humility. In recent years, we've begun to glimpse what cooperation with the sea might look like. Offshore wind farms harness its energy. Algae and seaweed farms offer sustainable food and biofuel. Marine protected areas allow ecosystems to rebound when left alone. Even the carbon stored in ocean sediments, what scientists call blue carbon, is being recognised as one of the planet's most effective forms of long-term climate storage. When we work with the ocean instead of against it, it responds. Life returns, currents heal, balance begins to restore itself. But more than solutions, what we need is perspective. To understand that we are not separate observers standing at the edge of the sea, but participants within it. Every breath we take connects us to phytoplankton drifting miles offshore. Every raindrop, every gust of wind, every meal we eat is a consequence of something the ocean set in motion long before us. It's easy to think of the ocean as vast beyond consequence, too big to harm, too powerful to change. But we've already changed it. And that realization should not lead to despair, but to understanding. Because if human activity can reshape the chemistry of a planet, then human intention can shape its recovery. We are, in a sense, the ocean becoming aware of itself. Through us, it studies its own patterns, names its own creatures, measures its own depth. We are its consciousness made temporary the brief moment when the planet looked inward and realized it was alive, and that awareness carries responsibility. To explore without taking, to understand without conquering, to exist within the same cycles that created us. If we can learn that lesson, the ocean will not just survive, it will continue to create, to balance, to remember. There is a quiet kind of hope in that idea. Not the hope of control, but of coexistence. Because for all our progress, we are still bound to the same physics, the same chemistry, the same flow of water that has governed Earth since the beginning. We will always be creatures of the tide. Someday, long after our cities have changed and our technologies have faded, the ocean will still be here moving, breathing, renewing. It will carry traces of us, just as it carries traces of the life that came before. And somewhere in its depths, future life will arise from what we leave behind. That is the way of the sea transformation without end. We began this journey with a paradox, that we have better maps of Mars than of our own ocean floor. Perhaps that's fitting. The ocean does not need us to map it to exist, it needs us to remember that we belong to it. When you stand at the shore and look out toward the horizon, you are not staring into emptiness. You're looking at the living memory of the world. You're looking at the reason you exist at all. The ocean is not Earth's last frontier, it is its first and its forever. If you enjoyed this journey, consider subscribing to Omni, where we explore the hidden stories of science, nature, and the universe. Every like, every comment, and every share helps these ideas reach more curious minds. Thanks for watching, and as always, stay curious. The largest living structure on Earth was built without blueprints, without machines, without any conscious plan. It stretches 2,300 kilometres along the Australian coast. Larger than Italy, visible from space, composed of 344,400 square kilometres of intricate architecture. The Great Barrier Reef contains roughly 2,900 individual reef systems, each one a masterwork of biological engineering that took thousands of years to construct, and every square centimetre was built by animals no bigger than your fingernail. For over a century, scientists assumed coral polyps simply extracted calcium and carbonate ions from seawater and deposited them directly onto their skeletons, like biological 3D printers working ion by ion. But in 2017, research published in the Proceedings of the National Academy of Sciences by Tali Maas and her team revealed something extraordinary. Corals don't build their skeletons through simple precipitation. They manufacture microscopic construction materials called amorphous calcium carbonate particles, transport these prefabricated components to the building site, and assemble them into crystalline aragonite structures. It's more sophisticated than any human construction process we've invented. According to studies published in Nature Communications in 2024, coral construction involves at least 5 distinct phases of mineral transformation, including previously unknown crystalline precursors like calcium carbonate hemihydrate that exist only during the building process. Researchers using advanced X-ray spectroscopy found that coral growth by particle attachment happens over 100 times faster than ion by ion precipitation from seawater, explaining how these tiny animals can create structures of such immense scale and complexity. But speed is only part of the story. Recent research in Advanced Materials in 2023 discovered that corals engineer their skeletons with deliberate structural imperfections, small misalignments between adjacent crystals that dramatically increase the material's toughness. This is why coral skeletons can support entire reef ecosystems weighing millions of tons while withstanding hurricanes, earthquakes, and constant wave action. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. moment a coral polyp begins construction, it faces an engineering challenge that would stump most human architects. Working in an environment where raw materials exist only as scattered ions dissolved in seawater, it must extract these components, concentrate them, transform them into solid building blocks, and assemble them into a structure that will last for centuries, all while continuing to feed, reproduce, and maintain its own life functions. Recent discoveries have revealed how corals accomplish this seemingly impossible task. Studies by Pupa Gilbert at the University of Wisconsin, published in Science in 2017, found that coral construction begins not at the skeleton surface, but deep inside the animal's tissues. Coral cells capture seawater in microscopic vesicles, concentrating calcium and carbonate ions to levels far higher than exist in the ocean. In these cellular factories, they manufacture amorphous calcium carbonate nanoparticles, roughly 400 nanometers in diameter, that serve as prefabricated building components. These particles aren't randomly produced. Research published in Crystal Growth and Design in 2022 showed that coral larvae begin manufacturing these construction materials even before they settle on the seafloor, preparing for skeleton formation while still floating in the water column. The process involves sophisticated quality control mechanisms mediated by specialized proteins called skeletal organic matrix proteins, which guide exactly how and where each particle crystallizes. The construction site itself is equally remarkable. Corals create specialized spaces called centers of calcification, microscopic workshops where the actual building occurs. As Mass and her colleagues discovered using high-resolution spectromicroscopy, these centers are enriched with organic fibers that serve as scaffolding, helping position and orient each nanoparticle as it transforms from amorphous calcium carbonate into crystalline aragonite. But perhaps most impressive is how corals coordinate construction across multiple scales simultaneously. Individual polyps, each no larger than a few millimeters, must synchronize their building efforts to create colonies that can span hundreds of meters. Different coral species have evolved distinct architectural strategies. Massive brain corals like Diploria labyrinthiformis grow slowly but create incredibly dense, sturdy foundations, extending only 0.3 to 2 centimeters per year according to research published in Coral Reefs Journal. Meanwhile, branching corals like Acropora cervicornis can grow up to 10 centimeters annually, rapidly extending reef frameworks into three-dimensional networks that maximize surface area for their symbiotic algae partners. The engineering becomes even more sophisticated when considering how corals adapt their construction to local conditions. Studies published in Coral Reefs in 2023 found that cold water corals automatically adjust their skeletal architecture based on current flow, growing more compact and dense in high-energy environments, more open and branching in calmer waters. The animals somehow sense mechanical stress and modify their building plans accordingly, creating structures optimized for their specific location. This adaptive construction happens without any central control system. Unlike human buildings that require architects, project managers, and coordinated teams, coral colonies achieve complex architectural goals through the simple principle that each polyp responds to local chemical and physical signals while following genetically encoded construction protocols. The result is architecture that emerges organically, optimized by millions of years of evolutionary refinement. Recent research has also revealed the incredible durability of coral construction. Studies show that healthy coral reefs can absorb up to 97% of wave energy, protecting coastlines more effectively than concrete seawalls while supporting ecosystems containing over 25% of all marine species. The secret lies in the multi-scale structure. At the nanoscale, organic matrix proteins create flexible interfaces between crystals. At the microscale, spherolytic crystal arrangements distribute stress. At the macroscale, the complex three-dimensional reef architecture dissipates wave energy through friction and turbulence. Tonight, we'll explore how these master builders perfect construction techniques that could revolutionize human architecture, why their building methods produce structures that are simultaneously stronger and more flexible than concrete, and what coral engineering reveals about the hidden intelligence that emerges when billions of simple components work together toward a common goal. For over a century, marine biologists operated under a simple assumption about how coral skeletons form. Coral polyps, they believed, worked like biological 3D printers, extracting calcium and carbonate ions directly from seawater and depositing them layer by layer onto their growing skeletons. The process seems straightforward. Seawater contains dissolved minerals, corals concentrate these minerals at the building site, and calcium carbonate crystallizes ion by ion from solution. This classical model made intuitive sense, but it had a problem. Coral skeletons don't match the chemical composition expected from direct precipitation from seawater. When researchers analysed the elemental ratios and isotopic signatures in coral aragonite, they found significant deviations from thermodynamic equilibrium, what scientists call vital effects. These chemical fingerprints suggested that something more complex was happening during skeleton formation, but nobody could figure out what. The breakthrough came when Tali Massa and her colleagues at the Weizmann Institute of Science decided to look inside living coral tissues using advanced spectromicroscopy techniques. What they discovered, published in the Proceedings of the National Academy of Sciences in 2017, overturned everything scientists thought they knew about coral construction. Corals don't precipitate their skeletons directly from seawater. Instead, they manufacture prefabricated building components deep within their tissues, transport these components to the construction site, and assemble them into crystalline structures through a sophisticated biological process that resembles modern industrial manufacturing more than traditional biological growth. The manufacturing process begins when coral cells capture seawater through a process called endocytosis, essentially swallowing small volumes of ocean water into specialized cellular compartments called vesicles. But these aren't passive storage containers. Once inside the vesicles, the coral cells actively modify the chemical environment, concentrating carbonate ions to levels far higher than exist in seawater, and creating conditions that favor the formation of amorphous calcium carbonate. Amorphous calcium carbonate, or ACC, is a peculiar material that exists in a state between liquid and solid. Unlike the familiar crystalline calcium carbonate found in limestone or mature coral skeletons, ACC lacks the ordered atomic structure of a true crystal. According to research published by Pupa Gilbert's team at the University of Wisconsin, ACC forms naturally when calcium and carbonate concentrations exceed certain thresholds, but it remains stable only under very specific conditions. Coral cells have learned to exploit this chemistry. Within their cellular vesicles, they manufacture two distinct types of ACC: hydrated amorphous calcium carbonate, which contains water molecules trapped within its structure, and anhydrous amorphous calcium carbonate, which contains no water. Each type has different properties and serves different construction purposes, though researchers are still unraveling exactly how corals control which type forms when. The manufacturing doesn't stop with ACC production. Mass and her colleagues found that coral cells produce these amorphous precursors as discrete nanoparticles, roughly 400 nanometers in diameter, about 200 times smaller than the width of a human hair. These aren't random blobs of material, but carefully manufactured construction components, with consistent size and composition. Once manufactured, these nanoparticles must be transported to where they're needed. Using high-resolution imaging techniques, researchers tracked the movement of ACC particles from their production sites, deep within coral tissues, to the spaces where skeleton formation occurs. The cells essentially operate a molecular-scale transportation system, moving construction materials through tissue layers to reach the building site. This discovery explains why coral construction happens so much faster than anyone expected. When Gilbert's team compared crystal growth rates, they found that skeleton formation through particle attachment occurs over 100 times faster than ion-by-ion precipitation from seawater. Instead of waiting for individual ions to randomly encounter the growing crystal surface, corals deliver preformed building blocks that can be rapidly integrated into the expanding skeleton. The speed advantage becomes even more remarkable when considering the scale of coral construction. A single large coral colony might contain millions of individual polyps, each manufacturing and deploying thousands of ACC nanoparticles every day. The collective output represents a massive biological construction project, with materials production, quality control, transport logistics, and assembly coordination all managed through cellular processes that operate without any central planning. But the nanoparticle manufacturing system also provides corals with unprecedented control over their construction materials. Unlike simple precipitation from seawater, which produces whatever crystal structure happens to be thermodynamically favoured, The ACC particle system allows corals to modify material properties before crystallization occurs. Research published in Nature Communications in 2024 by Sun, Mass, and their colleagues revealed that corals can tune the composition and structure of their ACC particles by adjusting the organic molecules present during manufacturing. These organic molecules, primarily proteins, lipids, and carbohydrates, don't just happen to be present during ACC formation. They're deliberately incorporated by coral cells as a form of molecular engineering. According to studies published in Crystal Growth and Design, different organic additives produce ACC particles with different crystallization behaviors. Some promote rapid transformation to aragonite, others slow it down, and still others influence the final crystal orientation and morphology. This means corals can engineer their construction materials at the molecular level, tailoring nanoparticle properties to match specific construction requirements. Areas of the skeleton that need high strength might receive ACC particles enriched with certain proteins, while regions requiring flexibility might get particles with different organic compositions. The coral essentially operates a custom materials manufacturing facility within every cell involved in skeleton formation. Recent research has revealed additional layers of sophistication in the ACC manufacturing process. Studies by Akiva and her colleagues published in Nature Communications in 2018 found that coral larvae begin producing ACC particles even before they settle on the seafloor and start building adult skeletons. This means coral construction capabilities are built into their developmental program from the earliest stages of life, with materials production systems coming online as soon as the animal begins forming hard structures. The precision of this manufacturing system becomes even more impressive when considering the chemical challenges corals face. Seawater chemistry varies with temperature, depth, season, and geographic location, but corals must consistently produce high-quality construction materials regardless of these fluctuations. Their cellular manufacturing systems include chemical sensors and regulatory mechanisms that maintain ACC particle quality across a wide range of environmental conditions. But manufacturing prefabricated components solves only part of the construction challenge. These ACC nanoparticles must somehow be assembled into the complex three-dimensional architectures that characterize coral skeletons. The particles don't magically organize themselves. They require guidance systems, assembly protocols, and quality control mechanisms that operate at scales ranging from nanometers to meters. That's where the real genius of coral engineering begins to emerge. The amorphous calcium carbonate nanoparticles that corals manufacture are just the beginning of a construction process that turns out to be far more complex than anyone imagined. For decades, scientists assumed that these ACC particles simply transform directly into the crystalline aragonite found in mature coral skeletons. One phase becomes another phase, end of story. Then, in 2024, researchers using a technique called myriad mapping, which analyzes millions of X-ray spectra simultaneously, made a startling discovery. Sun, Mass, and their colleagues, publishing in Nature Communications, found evidence for not just two phases in coral construction, but 5 distinct mineral phases working together in a carefully orchestrated sequence that resembles an industrial assembly line more than simple crystallization. The discovery came from examining freshly deposited coral skeleton surfaces with unprecedented resolution. Previous studies had missed the additional phases because they analysed only 3 components: the original amorphous precursors and the final crystalline product. But when the researchers expanded their analysis to search for 5 components, they found a dramatic improvement in how well their models fit the actual data. 2 previously unknown crystalline precursor phases were hiding in plain sight: calcium carbonate hemihydrate, abbreviated as CCHH, and monohydrocalcite, abbreviated as MHC. Both contain water molecules incorporated into their crystal structures, but in very specific arrangements that had never been observed in natural biominerals before. CCHH contains 1/2 water molecule per calcium carbonate unit, while MHC contains 1 full water molecule per unit. These crystalline precursors exist only briefly during the construction process, which explains why they were so difficult to detect. Unlike the relatively stable amorphous phases that can persist for hours, CCHH and MHC transform rapidly into aragonite, typically within minutes of formation. They appear to serve as intermediate steps in a carefully controlled crystallization pathway that gives corals exquisite control over the final crystal structure and orientation. The complete 5-phase construction process unfolds like this. Coral cells manufacture both hydrated and anhydrous amorphous calcium carbonate nanoparticles in their cellular vesicles. These particles are transported to the calcification site and deposited on the growing skeleton surface. Once in contact with the existing mineral framework, the amorphous particles don't immediately crystallize into aragonite. Instead, they undergo a series of solid-state transformations. First, the amorphous precursors partially organize into the crystalline intermediates CCHH and MHC. These phases have ordered atomic arrangements but retain significant amounts of water and maintain relatively high solubility compared to aragonite. During this intermediate phase, the coral can still modify crystal properties through organic matrix proteins that interact with the partially crystallized material. Next, the crystalline intermediates undergo progressive dehydration and atomic rearrangement to form progressively more stable calcium carbonate phases. According to thermodynamic calculations published alongside the 2024 study, this creates a complex energy landscape with multiple possible transformation pathways, allowing corals to fine-tune which route their materials follow based on local conditions and construction requirements. Finally, all precursor phases transform into mature aragonite crystals with the specific orientations and morphologies needed for that particular location in the skeleton. The entire process typically takes about 24 hours from initial particle deposition to complete crystallization, though the timing can be adjusted based on environmental conditions and construction demands. What makes this process remarkable is not just its complexity, but how corals control each step through biological mechanisms. Research published in Science in 2017 by Mass and her colleagues found that organic matrix proteins secreted by coral cells don't just passively template crystal formation. They actively guide the transformation pathway by stabilizing certain intermediate phases while destabilizing others. Different coral species appear to use different combinations of these transformation pathways. Studies comparing coral skeletons from 13 different species found that while all mature coral skeletons consist of aragonite, arranged in spherulitic patterns, the intermediate phases and transformation routes vary significantly between species. This suggests that corals have evolved multiple engineering approaches to achieve similar structural goals, much like how human engineers might use different manufacturing processes to produce parts with similar specifications. The discovery of crystalline intermediate phases also resolves a long-standing puzzle about coral construction energetics. Transforming directly from amorphous calcium carbonate to crystalline aragonite requires overcoming a substantial energy barrier, which should make the process slow and inefficient. But by routing construction through intermediate crystalline phases, corals create a stepwise transformation pathway with much lower energy barriers at each step. This staged approach explains how corals can build skeletons so rapidly while maintaining precise control over crystal properties. Instead of forcing materials to make a dramatic phase transition all at once, they guide particles through a series of smaller, more manageable transformations. Each step provides an opportunity for biological control mechanisms to adjust crystal properties, correct errors, or redirect the transformation based on local construction requirements. The intermediate phases also provide a quality control mechanism that's impossible with direct crystallization from solution. If an ACC particle contains impurities or has improper composition, the intermediate crystalline phases give coral cells time to detect and correct these problems before the material locks into its final aragonite structure. Defective particles can be dissolved and recycled, while properly formed ones continue through the transformation sequence. Recent research has revealed that corals can even pause the transformation process when necessary. Under stressful conditions like elevated temperature or reduced pH, corals can stabilize particles in intermediate phases rather than completing crystallization. This allows them to maintain construction readiness without expending the full energy cost of creating mature skeleton, essentially creating a construction material stockpile that can be rapidly deployed when conditions improve. The 5-phase construction process also explains why coral skeletons have such unusual mechanical properties. Traditional crystallization from solution tends to produce materials with relatively predictable structures and properties determined primarily by the chemical composition. But the stepwise transformation pathway allows corals to engineer crystal defects, grain boundaries, and other structural features that dramatically improve material performance. Understanding this construction process has profound implications for how we think about biological engineering more generally. Corals have evolved what amounts to a sophisticated materials processing system that would be challenging to replicate even with modern industrial technology. They manufacture raw materials, control transformation pathways, implement quality control systems, and optimize final product properties, all using cellular machinery operating at ambient temperature and pressure. But even the most sophisticated materials processing system is useless without coordination mechanisms that ensure individual construction efforts combine to create coherent large-scale structures. Millions of coral polyps must somehow synchronize their 5-phase construction processes to build unified skeletons that can span hundreds of meters and persist for centuries. How do these tiny animals, each following their own local construction protocols, manage to create architectural masterpieces that dwarf anything humans have ever built? The challenge facing a coral colony is unlike anything human engineers have ever attempted. Imagine trying to construct a cathedral using thousands of independent contractors, each working on their own small section, with no blueprints, no project manager, and no communication system except chemical signals that diffuse slowly through water. Each worker must somehow coordinate their efforts with neighbours they can't see, following construction rules they've never been taught, to create a unified structure that will stand for centuries. This is exactly what coral colonies accomplish every day, and recent research has begun to reveal how they do it. The key lies in specialised construction sites called centres of calcification, or COCs. These microscopic workshops, discovered through high-resolution imaging studies by Mass and her colleagues, serve as the focal points where coral skeleton formation actually occurs. According to research published in Science in 2017, each coral polyp operates multiple COCs simultaneously, typically arranged in species-specific patterns that reflect the animal's underlying body plan. Within each centre of calcification, coral cells secrete an intricate network of organic fibres composed primarily of proteins, lipids, and carbohydrates. This organic matrix doesn't just provide a surface for crystal nucleation, it functions as a sophisticated guidance system that controls exactly how and where each amorphous calcium carbonate nanoparticle transforms into crystalline aragonite. Studies published in Advanced Materials in 2023 reveal that different coral species produce dramatically different organic matrix compositions, each optimized for their particular architectural strategy. Brain corals like Diploria labyrinthiformis create matrix proteins that promote the formation of dense, tightly packed crystal arrangements, producing the massive boulder-like structures that form reef foundations. Branching corals like Acropora cervicornis manufacture matrix proteins that guide crystals to grow in elongated, radiating patterns that create the delicate, tree-like architectures that maximize surface area for their symbiotic algae. But the most remarkable discovery is how coral colonies coordinate construction across multiple polyps to create coherent large-scale structures. Research by Gilbert and her team, using a technique called polarization-dependent imaging contrast mapping, found that adjacent coral polyps somehow align their crystal growth directions with extraordinary precision. In healthy coral, colonies, aragonic crystals from neighbouring polyps orient within just a few degrees of each other, creating seamless structural continuity across the entire skeleton. This coordination happens without any apparent communication mechanism between polyps. Individual coral animals lack nervous systems sophisticated enough to coordinate complex construction projects, and they're separated by their own skeletons, which should prevent direct chemical communication. Yet somehow, colonies achieve architectural precision that would challenge human construction teams equipped with laser surveying equipment and computer-aided design systems. The solution, according to recent research, lies in a phenomenon called constrained crystallization. When coral polyps deposit their amorphous calcium carbonate nanoparticles onto existing skeleton surfaces, the particles don't crystallize randomly. Instead, they're influenced by the crystal orientations already present in the underlying structure. The existing skeleton essentially serves as a template that guides new crystal formation, ensuring that additions align properly with what's already been built. This process works because of the 5-phase construction pathway discovered in 2024. The intermediate crystalline phases, calcium carbonate, hemihydrate, and monohydrocalcite, remain sensitive to structural influences from surrounding crystals much longer than if corals crystallized aragonite directly from solution. During the hours-long transformation process, organic matrix proteins can sense the orientations of nearby crystals and guide new particle transformations to match. Studies published in Proceedings of the National Academy of Sciences in 2021 revealed additional layers of sophistication in this coordination system. Coral colonies don't just maintain crystal alignment, they actively optimize their large-scale architecture based on environmental conditions. Colonies growing in high-energy wave environments automatically develop more compact, streamlined shapes, while those in calmer waters create more branching, three-dimensional structures that maximize light capture for photosynthesis. This environmental responsiveness emerges from the interaction between local construction rules and colony-wide feedback systems. Individual polyps can sense mechanical stress through their connection to the shared skeleton, and they modify their construction behavior accordingly. Polyps experiencing high wave forces produce denser, more robust skeletal elements, while those in protected areas can afford to build more delicate, efficient structures. The coordination system also includes error correction mechanisms that maintain architectural integrity, even when individual polyps make construction mistakes or die. Research published in Coral Reefs in 2019 found that coral colonies can detect and repair structural weaknesses by redirecting construction resources to problem areas. Neighbouring polyps somehow sense when skeleton formation is disrupted and increase their own construction rates to compensate. Perhaps most impressively, coral colonies can adapt their architectural strategies over time as they grow. Young colonies typically focus on rapid vertical growth to compete for light and space, producing relatively simple, pillar-like structures. As colonies mature and establish dominance in their local environment, they shift to more complex branching patterns that maximize surface area and create habitat for other organisms. This architectural flexibility emerges from genetic programs that govern how organic matrix proteins are produced and deployed during different life stages. Studies of coral gene expression by researchers at the Australian Institute of Marine Science found that colonies actively regulate the production of different matrix protein types based on their size, age, and environmental conditions. Larger colonies express different suites of construction-related genes than smaller ones, effectively switching their architectural programming as they grow. The resulting structures exhibit a mathematical elegance that suggests deep, evolutionary optimization. Coral skeletons consistently display fractal-like properties, where similar branching patterns repeat at different scales from millimeters to meters. This fractal architecture maximizes surface area while minimizing material usage, creating structures that are simultaneously efficient and robust. Recent computational studies have confirmed that coral architectural patterns closely approximate mathematical solutions to optimization problems in structural engineering. When researchers use computer algorithms to design structures that maximize surface area while minimizing material costs and maintaining structural stability, the results closely resembled the branching patterns found in real coral colonies. But corals achieve these mathematically optimal designs without computers, calculations, or conscious planning. The patterns emerge naturally from the interaction between simple local rules followed by individual polyps and the physical constraints imposed by their environment. Each polyp follows genetically encoded instructions about where and how to deposit construction materials, but the large-scale architectural outcome emerges from the collective behavior of millions of these simple rule followers. This is a profound example of what scientists call emergent intelligence, complex, apparently purposeful behavior that arises from the interaction of many simple components, none of which understands the overall system. The coral colony as a whole exhibits sophisticated architectural decision-making, but this intelligence doesn't reside in any single location or component. Instead, it emerges from the dynamic interaction between construction rules, environmental feedback, and the physical properties of the materials themselves. Understanding this emergent coordination system has implications far beyond coral biology. Engineers and architects are beginning to explore how similar principles might be applied to human construction projects, particularly in situations where traditional centralized planning approaches are impractical or impossible. But even the most sophisticated coordination system is only as good as the materials it works with. The architectural mastery that coral colonies display depends fundamentally on the extraordinary properties of the materials they construct, properties that are only now beginning to be understood and that challenge everything engineers thought they knew about optimal structural design. For most of human history, engineers have pursued a simple principle: perfection equals performance. The strongest steel contains the fewest impurities, the most durable concrete maintains uniform composition, and the best crystals display flawless atomic arrangements. Structural defects, misalignments, and irregularities represent failures to be minimized or eliminated entirely. Corals follow exactly the opposite philosophy, and recent research reveals why their approach produces superior results. The breakthrough came when James Schwenk and Pupa Gilbert at the University of Wisconsin began examining coral skeletons with a technique called polarization-dependent imaging contrast mapping, which can reveal the orientations of individual crystals within complex materials. What they found, published in Advanced Materials in 2023, overturned fundamental assumptions about optimal structural design. Coral skeletons aren't built from perfectly aligned crystals. Instead, they consist of what scientists call spherulites, radiating clusters of needle-like aragonite crystals that grow outward from common centers, with each crystal slightly misoriented relative to its neighbors. The misalignment isn't random or accidental. It follows precise mathematical relationships that optimize the material's mechanical properties in ways that perfect crystal alignment never could. In a perfectly crystalline material, cracks propagate easily along crystal boundaries because the atomic planes provide natural fracture pathways. But in coral spherulites, the deliberate misalignments between adjacent crystals force cracks to constantly change direction as they encounter differently oriented crystal faces. This crack deflection dramatically increases the energy required to break the material, making coral skeletons far tougher than would be possible with perfect crystal alignment. The discovery explains a long-standing puzzle about coral engineering. Aragonite, the mineral that comprises coral skeletons, has relatively modest mechanical properties when grown as perfect single crystals in laboratory conditions. But coral skeletons demonstrate exceptional toughness, flexibility, and resistance to fracture, properties that seemed inconsistent with their chemical composition. The secret lies not in the material itself, but in how corals organize it at the nanoscale. Gilbert's team found that the degree of crystal misalignment varies systematically throughout coral skeletons based on functional requirements. Areas that experience high mechanical stress contain crystals with larger misorientation angles, maximizing toughness at the expense of some stiffness. Regions that require structural rigidity maintain smaller misalignment angles, optimizing stiffness while preserving adequate fracture resistance. This means corals actively tune their material properties at the atomic level by controlling crystal organization during construction. The organic matrix proteins secreted within centers of calcification don't just guide crystal nucleation, they precisely control the angular relationships between adjacent crystals to optimize mechanical performance for local conditions. Recent research published in Nature Materials in 2024 revealed additional sophistication in coral crystal engineering. Using advanced X-ray diffraction techniques, researchers found that coral spherulites contain systematic gradients in crystal properties from their centers to their edges. Crystals near spherulite centers tend to be smaller and more heavily misoriented, providing flexibility and energy absorption. Crystals toward the edges are larger and more aligned, contributing structural stiffness and load-bearing capacity. These property gradients create a hierarchical toughening mechanism that operates across multiple length scales simultaneously. At the nanoscale, organic molecules between crystal interfaces provide flexibility. At the microscale, crystal misalignments deflect cracks. At the mesoscale, spherulite arrangements distribute loads efficiently. At the macroscale, the overall skeletal architecture channels forces along optimal pathways. The result is a material that performs better than human-engineered composites that cost thousands of times more to manufacture. Mechanical testing by researchers at MIT found that coral skeletons exhibit specific toughness values, the energy required to fracture a unit mass of material, that rival or exceed those of modern fiber-reinforced composites while maintaining the ability to self-repair through continued biological growth. But coral crystal engineering goes beyond just optimizing mechanical properties. Studies published in Science in 2017 revealed that coral spherulitic structures also enhance the optical properties of skeletal elements. The radiating crystal arrangements scatter and redirect light in ways that improve photon delivery to the symbiotic algae living within coral tissues, effectively turning the skeleton into a biological optical system. This dual optimization, simultaneously maximizing mechanical performance and optical efficiency, represents a level of materials engineering sophistication that human technology struggles to match. Current human composites excel at either mechanical properties or optical properties, but rarely both. Achieving dual optimization typically requires complex manufacturing processes, exotic materials, or trade-offs that compromise one property to enhance another. Corals achieve both simultaneously, using only calcium carbonate and seawater, assembled at ambient temperature and pressure through biological processes. The key lies in their precise control over crystal nucleation, growth, and organization through organic matrix proteins that have been refined by hundreds of millions of years of evolutionary optimization. Recent computational studies have confirmed that coral crystal arrangements closely approximate mathematical solutions to multi-objective optimization problems in materials science. When researchers used advanced algorithms to design crystal structures that simultaneously maximize toughness, stiffness, and optical transmission while minimizing material usage, the results closely resembled the spherulitic patterns found in real coral skeletons. The implications extend far beyond understanding coral biology. Material scientists are now attempting to recreate coral-inspired crystal organization in synthetic materials, hoping to develop new composites that match coral performance characteristics. Early results are promising, but highlight the extraordinary sophistication of biological crystal engineering. Attempts to grow synthetic aragonite spherulites with coral-like properties require careful control over solution chemistry, temperature, pH, organic additives, and nucleation timing, parameters that corals manage automatically through cellular machinery. Even with precise laboratory control, synthetic spherulites rarely achieve the structural perfection and property optimization displayed by biological examples. The challenge illustrates how evolution has solved materials engineering problems that remain at the frontiers of human technology. Coral polyps, working with genetically encoded instructions refined over geological timescales, routinely produce materials with performance characteristics that challenge the best human engineers equipped with sophisticated analytical tools and manufacturing capabilities. But perhaps most remarkably, corals achieve this materials engineering mastery while simultaneously solving an even greater challenge that no human engineer has ever faced. They must construct these high-performance structures not in controlled factory conditions, but in one of the most chemically aggressive environments on Earth. Seawater that is simultaneously trying to dissolve their skeletons, organisms that are actively boring through their structures, and wave forces that can exceed those experienced by offshore oil platforms. Building superior materials is impressive. Building them in conditions that would destroy most human-engineered structures represents a level of engineering sophistication that challenges everything we understand about construction in hostile environments. Imagine trying to construct a skyscraper while the foundation continuously dissolves beneath you. The building materials corrode in transit, and demolition crews work around the clock to tear down everything you build. Add hurricane-force winds that strike without warning, and chemical attacks that make concrete brittle and steel rust within hours. No human engineer would attempt construction under such conditions, yet this describes the normal operating environment for coral polyps. Seawater represents one of the most chemically aggressive construction environments on Earth. It contains dissolved salts that corrode metals, acids that dissolve carbonates, and microorganisms that bore through solid rock. Ocean chemistry fluctuates daily with temperature changes, seasonally with biological cycles, and over decades with climate shifts. Wave forces can exceed those experienced by offshore oil platforms, subjecting structures to constant mechanical stress punctuated by catastrophic storm events. For organisms that build skeletons from calcium carbonate, one of the most chemically vulnerable construction materials possible, these conditions should make large-scale construction impossible. Calcium carbonate dissolves readily in acidic water, becomes mechanically weak when hydrated, and provides an attractive target for boring organisms that tunnel through coral skeletons for shelter and food. Yet corals have not merely survived in this hostile environment, they have thrived, creating the largest biological structures on Earth while maintaining construction operations for over 500 million years. The secret lies in a suite of engineering adaptations that would be impossible to implement in human construction projects. Recent research has revealed how corals modify their building strategies, materials, and architectural approaches based on local environmental conditions, creating structures optimized for the specific challenges they face. Studies published in Coral Reefs in 2023 by researchers examining cold-water coral construction found that species living in high-energy environments automatically produce more compact, streamlined skeletal architectures. Desmophyllum pertusum colonies growing in strong currents develop volume compactness values, a measure of how efficiently a structure uses space, that are significantly higher than those growing in calmer waters. The corals somehow sense flow conditions and adjust their construction accordingly, creating architectures that minimize drag while maintaining structural integrity. This environmental responsiveness operates at multiple scales simultaneously. At the molecular level, corals adjust the composition of their organic matrix proteins based on water chemistry conditions. Research published in Science in 2017 by Paul Falkowski's team at Rutgers University found that corals use highly acidic proteins to catalyze calcium carbonate formation, allowing them to continue building even as ocean acidification makes carbonate precipitation thermodynamically unfavorable. The acidic proteins work by creating local chemical microenvironments around construction sites, where carbonate formation remains possible even when the surrounding seawater chemistry would normally prevent crystallization. This represents a sophisticated form of chemical engineering that allows corals to maintain construction operations despite deteriorating ocean chemistry. At the cellular level, corals implement quality control systems that detect and repair construction defects caused by environmental stress. Studies of coral construction under elevated temperature conditions found that polyps can pause crystal formation processes when conditions become unfavorable, essentially putting construction projects on hold until environmental conditions improve rather than producing inferior materials. When construction does resume, corals implement what researchers call catch-up growth strategies that rapidly restore skeletal integrity. Research published in the Proceedings of the National Academy of Sciences in 2021 found that coral colonies can increase their construction rates by up to 300% following stress events, allocating additional metabolic resources to skeleton formation until structural deficits are corrected. But perhaps the most impressive adaptation is how corals engineer their structures to actually benefit from environmental forces that would destroy human constructions. Wave energy, which represents a major threat to artificial structures, becomes a resource that corals exploit to enhance their construction operations. Studies published in Coral Reefs in 2019 found that moderate wave action actually increases coral calcification rates by up to 40% compared to calm water conditions. The mechanical energy from waves enhances mixing around coral polyps, improving the delivery of construction materials and the removal of metabolic waste products. Corals have learned to use environmental forces as part of their construction infrastructure. However, corals face an escalating challenge that no previous generation of reef builders encountered: rapidly changing ocean chemistry driven by human carbon dioxide emissions. Ocean acidification reduces the concentration of carbonate ions that corals need for skeleton formation, while simultaneously increasing the solubility of existing skeletal structures. Research published in Nature Climate Change in 2023 found that many coral reefs now exist in seawater conditions that are marginal for carbonate construction. Some reefs experience daily fluctuations in carbonate chemistry that swing from barely suitable for construction during daylight hours to actively corrosive at night. Under these conditions, coral polyps must not only build new skeleton during favourable periods, but also repair dissolution damage that occurs during unfavourable periods. Corals have responded by developing more sophisticated construction scheduling strategies. Studies of coral calcification patterns found that many species now concentrate their skeleton building activities during narrow windows, when carbonate chemistry conditions are optimal, essentially operating construction shifts that align with daily chemical cycles. Some coral species have gone further modifying their skeletal architecture to reduce vulnerability to dissolution. Research comparing historical and contemporary coral specimens found that many modern corals produce skeletons with higher concentrations of organic matrix proteins, creating materials that are more resistant to acid dissolution even if they require more energy to manufacture. The bioerosion challenge represents another escalating threat that corals must engineer around. Parrotfish, boring sponges, bioeroding worms, and other organisms actively destroy coral skeletons, removing calcium carbonate as fast as corals can deposit it. On heavily impacted reefs, bioerosion rates can exceed coral construction rates, leading to net skeleton loss even from healthy, actively growing colonies. Corals have evolved several strategies to combat bioerosion. Some species produce chemical deterrents that discourage boring organisms, while others create skeletal architectures that are mechanically difficult to penetrate. Research published in Marine Biology in 2022 found that coral species subject to intense bioerosion pressure tend to evolve denser, more compact skeletal structures that resist mechanical boring. Most impressively, some corals have learned to use bioerosion to their advantage. Studies of coral reef dynamics found that moderate levels of bioerosion actually enhance overall reef construction by creating spaces that are rapidly colonized by young corals and other calcifying organisms. The bioeroders essentially perform biological renovation, preventing reefs from becoming overgrown with dead coral skeleton, while creating settlement opportunities for new construction. Climate change has added another layer of complexity to coral construction challenges. Rising sea temperatures increase coral metabolic rates, potentially accelerating construction, but they also increase the frequency and severity of thermal stress events that disrupt construction entirely. More frequent storms increase mechanical damage rates, while changing precipitation patterns alter coastal water chemistry. Coral responses to these changing conditions provide insights into the flexibility of biological construction systems. Research published in Global Change Biology in 2023 found that coral species with more flexible construction strategies, those capable of adjusting their architectural approaches, materials properties, and construction timing, show greater resilience to environmental change than species with more rigid construction programs. The most successful reef-building corals appear to be those that treat construction not as a fixed biological process, but as an adaptive response that can be tuned based on environmental conditions. They modify their engineering approaches continuously, optimizing their construction strategies for whatever conditions they encounter, rather than following predetermined building programs. This adaptive flexibility represents a fundamental difference between biological and human engineering approaches. Human construction projects typically involve detailed planning phases that specify materials, methods, and schedules before construction begins. Changes to these plans are expensive and disruptive, creating systems that perform well under anticipated conditions but struggle to adapt when conditions change. Coral construction operates more like an improvisational jazz performance than a scripted architectural project. Individual polyps follow basic construction principles but continuously adjust their implementation based on real-time environmental feedback. The result is construction systems that remain functional across a wide range of conditions while continuously optimizing their performance. Understanding how corals maintain construction operations under such challenging conditions is providing insights that could revolutionize human approaches to building in harsh environments. From offshore platforms that must withstand marine corrosion to infrastructure in chemically aggressive industrial settings, coral engineering principles offer potential solutions to some of our most challenging construction problems. But perhaps the most profound lesson from coral construction is the power of scaling simple solutions across enormous systems. Individual coral polyps may be tiny, but when millions of them coordinate their construction efforts over decades or centuries, they create structures that dwarf anything humans have ever built. A single coral polyp measures perhaps 2 millimeters across, smaller than the head of a pin. Its skeleton construction capabilities extend maybe 10 centimeters beyond its body during an entire year of growth. By any reasonable measure, an individual coral represents one of nature's most modest construction projects, building structures that a human could span with outstretched fingers. Yet somehow, through processes that challenge everything engineers understand about scaling construction projects, these microscopic builders create structures that reshape entire continents. The Great Barrier Reef stretches 2,300 kilometres along Australia's northeast coast, covering 344,400 square kilometres of ocean, an area larger than Italy, Germany, or the state of New Mexico. The reef system contains approximately 2,900 individual reefs and 900 islands, representing roughly 10% of all coral reef ecosystems on Earth. According to studies published by the Great Barrier Reef Marine Park Authority in 2019, the reef supports over 9,000 species of marine life and contributes approximately $6.4 billion annually to Australia's economy. Every square centimetre of this continental scale architecture was constructed by animals smaller than your thumb. Understanding how coral construction scales from individual polyps to planetary scale structures requires grappling with mathematics that operate across 12 orders of magnitude, from nanometer scale crystal formation to 1,000-kilometre reef systems. Recent research has begun to reveal how this extraordinary scaling becomes possible through principles that could revolutionise human approaches to large-scale construction projects. The scaling begins with time. Individual coral polyps may build slowly, but they never stop building. Research published in Coral Reefs in 2019 found that healthy coral colonies maintain continuous skeleton formation 24 hours a day, 365 days a year, for decades or centuries. A brain coral colony in the Caribbean can live for over 900 years, depositing calcium carbonate continuously throughout its entire lifespan. The cumulative effect of this relentless construction creates structures that grow from millimeters to meters over human lifetimes. But individual colony growth represents only the first level of scaling. Coral reefs emerge when thousands of individual colonies coordinate their construction efforts across spatial scales ranging from meters to hundreds of kilometers. Studies published in Nature in 2020 found that successful reef formation requires what researchers call recruitment cascades, processes where the construction success of existing coral colonies creates conditions that facilitate the establishment and growth of new colonies. The mechanism works through habitat modification. As coral colonies grow, they create three-dimensional structures that alter local water flow, sediment transport, and light conditions. These modifications create microenvironments that are more favourable for coral larval settlement and growth. Young corals that establish successfully in these modified environments further alter local conditions, creating positive feedback loops that facilitate even more coral recruitment. Over decades, these recruitment cascades transform flat seafloor into complex three-dimensional reef structures. Research published in the Proceedings of the National Academy of Sciences in 2021 found that mature coral reefs can rise 50 to 100 meters above the surrounding seafloor, creating underwater mountains built entirely through biological construction processes. The vertical growth rates, while individually modest, become impressive when aggregated across entire reef systems. Studies of Holocene reef development found that healthy coral reef systems can maintain vertical accretion rates of 8 to 10 millimeters per year over periods of thousands of years. This means reef systems can keep pace with sea level rise, maintain their position in the optimal depth range for coral growth, and continue expanding even as geological processes gradually subside the underlying seafloor. But the most remarkable aspect of coral reef scaling lies in how construction decisions made by individual polyps propagate to influence ecosystem-level properties. Research published in Scientific Reports in 2017 found that the architectural complexity of coral reefs, measured as the amount of three-dimensional structure created per unit area, directly determines the reef's capacity to support marine biodiversity. Reefs with more complex architecture provide more niches for different species, more shelter from predation, and more efficient nutrient cycling. Studies comparing reefs with different architectural complexity found that small increases in structural complexity can support exponentially more species. This means that construction decisions made by individual coral polyps, choices about skeletal morphology, growth direction, and colony architecture directly influence the ecological functions of entire ecosystems. The scaling effects extend beyond individual reef systems to influence regional and global oceanic processes. Research, published in Global Biogeochemical Cycles in 2022, found that coral reef construction represents a major component of the global carbon cycle. with reef systems collectively sequestering approximately 70 to 80 million tons of carbon dioxide annually in the form of calcium carbonate skeletons. This carbon sequestration occurs through what scientists call the carbonate counterpump, a process where coral construction removes dissolved inorganic carbon from seawater, allowing the ocean to absorb more carbon dioxide from the atmosphere. The magnitude of this effect depends on the total construction activity across all coral reef systems globally, meaning that construction decisions by individual polyps contribute to planetary-scale climate regulation. Recent studies have revealed additional ways that microscopic coral construction influences planetary-scale processes. Research published in Nature Geoscience in 2023 found that coral reef construction creates distinctive signatures in seawater chemistry that can be detected thousands of kilometers away from reef systems. As coral colonies extract calcium and carbonate from seawater for skeleton formation, they alter the chemical composition of surrounding water masses. These chemical modifications persist as water masses circulate through ocean basins, creating traceable fingerprints of coral construction activity. The chemical signatures provide insights into how biological construction processes influence ocean chemistry on geological timescales. Studies of marine sediment cores found evidence that massive coral reef construction events in Earth's past created chemical changes that persisted for millions of years, influencing the evolution of seawater composition and the availability of construction materials for future reef-building organisms. Perhaps most remarkably, coral construction creates emergent properties that cannot be predicted from understanding individual polyp behavior. Research published in Ecological Monographs in 2021 found that large coral reef systems exhibit self-organizing properties that optimize their structural complexity, species diversity, and ecological functions without any centralized control mechanisms. These emergent properties arise through what complexity scientists call scale-free networks, organizational patterns where small-scale components interact through simple rules to create large-scale structures that exhibit consistent statistical properties regardless of system size. Coral reefs display mathematical relationships between construction activity, architectural complexity, and biodiversity that follow power law distributions spanning multiple orders of magnitude. The scale-free properties explain how coral construction systems remain stable and functional even as individual colonies grow, die, and are replaced over time. The statistical relationships between different components of the reef system create self-regulating feedback mechanisms that maintain overall system properties even as individual components change. Understanding these scaling principles has profound implications for human construction projects. Most human engineering approaches rely on centralized planning and hierarchical control systems that become increasingly difficult to manage as project size increases. Large construction projects require extensive coordination, complex logistics, and sophisticated management systems that often fail when projects exceed certain size thresholds. Coral construction suggests alternative approaches based on distributed decision-making, local optimization, and emergent coordination. Instead of trying to plan and control every aspect of large construction projects, coral-inspired approaches might focus on establishing simple rules and feedback mechanisms that allow construction systems to self-organize toward optimal configurations. Early experiments with coral-inspired construction approaches are showing promising results. Research published in Nature Materials in 2024 found that construction systems based on coral organizational principles could create complex structures using simple robots following local interaction rules without centralized planning or global knowledge of the construction project. But the most profound lesson from coral scaling may be about time itself. Human construction projects typically operate on timescales of months to decades, with success measured by completion within predetermined schedules and budgets. Coral construction operates on timescales of centuries to millennia, with success measured by persistence and adaptation over geological time periods. This temporal perspective suggests that truly sustainable construction might require abandoning human concepts of project completion in favor of approaches based on continuous adaptation and indefinite maintenance. Rather than building structures designed to last for specific periods, coral-inspired approaches might focus on creating construction systems that can continuously modify and repair themselves as conditions change. The scaling from polyp to planet represents perhaps the most extraordinary example of emergent complexity in the natural world. Individual organisms following simple genetically encoded construction rules somehow create planetary-scale structures that influence ocean chemistry, global carbon cycles, and the evolution of marine ecosystems. But this ancient mastery, refined over 500 million years of evolutionary optimization, now faces challenges that no previous generation of reef builders has ever encountered. In 2016, aerial surveys revealed that 29% of corals in Australia's Great Barrier Reef had died within a single year, victims of ocean temperatures that exceeded coral thermal tolerance by just 1 to 2 degrees Celsius. The event marked a turning point in the 500 million year history of coral reef construction, the first time that human activities had begun outpacing the ability of coral engineering systems to adapt and recover. The mathematics of the threat are sobering. Ocean acidification, driven by increasing atmospheric carbon dioxide, is reducing the concentration of carbonate ions that corals need for skeleton formation, while simultaneously increasing the solubility of existing coral structures. Research published in Nature Climate Change in 2023 found that by 2050, seawater chemistry conditions at many coral reef locations will make calcium carbonate construction thermodynamically unfavourable for significant portions of each day. Rising sea temperatures compound the chemical challenges by disrupting the symbiotic relationships between corals and their algal partners that power construction processes. Studies published in Science in 2021 found that coral bleaching events, episodes where thermal stress causes corals to expel their algae, now occur with such frequency that many coral colonies lack sufficient recovery time between stress events to rebuild their skeletons and energy reserves. But perhaps most critically, the pace of environmental change now exceeds the timescales on which coral construction systems operate. Coral colonies that evolved to optimize their building strategies over decades or centuries suddenly face environmental conditions that change from year to year. Construction systems that relied on environmental stability to achieve their remarkable scaling properties struggle to function in a world of accelerating change. Yet even as coral reefs face unprecedented challenges, scientists are discovering that coral construction principles offer revolutionary insights for human engineering and architecture. The race to understand coral building technologies has become not just a matter of biological conservation, but a quest to unlock construction capabilities that could transform how humans approach building in an uncertain future. Research published in Nature Materials in 2024 found that coral-inspired construction approaches could enable human buildings to adapt continuously to changing environmental conditions rather than failing catastrophically when conditions exceed design parameters. Instead of constructing rigid structures designed for specific conditions, coral-inspired architecture would create buildings that continuously modify their properties, repair damage automatically, and optimize their performance as conditions change. The key insight lies in coral approaches to materials engineering. While human construction typically relies on materials with fixed properties determined during manufacturing, coral construction creates materials that can modify their properties continuously throughout their service life. Studies published in Advanced Materials in 2023 found that coral skeletons can adjust their stiffness, strength, and toughness in response to changing mechanical loads, essentially tuning their material properties to match functional requirements. Researchers at MIT are now developing synthetic materials inspired by coral organic matrix systems that can modify their crystal structures in response to environmental stimuli. Early prototypes demonstrate concrete-like materials that become stronger when exposed to acidic conditions rather than degrading, mimicking how corals use acidic proteins to maintain construction capabilities even under chemically challenging conditions. The construction industry is also exploring coral-inspired approaches to large-scale project coordination. Traditional construction projects rely on centralized planning and hierarchical control systems that become increasingly fragile as project size increases. Research published in Nature Communications in 2023 found that construction systems based on coral organizational principles, where individual components follow simple local rules that create complex global behaviors, could potentially manage construction projects of unprecedented scale and complexity. Early experiments with swarm construction systems demonstrate how hundreds of simple robots following coral-inspired interaction rules can create complex three-dimensional structures without centralized planning or global knowledge of the construction project. The robots essentially replicate coral polyp behavior, making local construction decisions based on their immediate environment while collectively creating coherent large-scale architectures. Perhaps most intriguingly, researchers are exploring how coral construction principles might enable the development of truly living architecture, buildings that grow, adapt, and repair themselves through biological processes. Studies published in Science Advances in 2024 found that engineered microorganisms could potentially be programmed to secrete calcium carbonate in controlled patterns, creating building materials that literally grow themselves while adapting to local environmental conditions. These bioarchitectural approaches could enable construction in environments where traditional building methods are impractical or impossible. Underwater structures, buildings in chemically aggressive industrial environments, or construction projects in remote locations could potentially be grown rather than built, using local materials and energy sources while adapting continuously to site-specific conditions. The implications extend beyond engineering to fundamental questions about the nature of intelligence and problem-solving in biological systems. Coral construction represents perhaps the purest example of what scientists call distributed intelligence, sophisticated problem-solving that emerges from the interaction of many simple components, none of which understands the overall system. Research published in Proceedings of the National Academy of Sciences in 2021 found that coral construction systems solve optimization problems that challenge the most sophisticated human algorithms, but they do so through evolutionary processes rather than conscious design. The mathematical elegance of coral architectures, the precision of their materials engineering, and the sophistication of their scaling strategies all emerge from genetic programs refined over hundreds of millions of years. This suggests that biological systems have access to problem-solving capabilities that transcend individual consciousness or intelligence. Coral colonies exhibit architectural decision-making that appears purposeful and sophisticated, but this intelligence doesn't reside in any single location or component. Instead, it emerges from the dynamic interaction between construction rules, environmental feedback, and the accumulated wisdom of evolutionary optimization. Understanding these distributed intelligence systems could revolutionize human approaches to complex problem-solving more generally. Instead of relying on centralized analysis and planning, coral-inspired approaches might harness collective intelligence through systems where many simple decision-makers interact according to carefully designed rules. But perhaps the most profound lesson from coral construction lies in its temporal perspective. Human engineering typically focuses on creating solutions to immediate problems, with success measured by functionality within predetermined timeframes. Coral construction operates on geological timescales, creating solutions that persist and adapt across millions of years of environmental change. This temporal perspective suggests that truly sustainable engineering might require fundamental shifts in how humans conceptualize construction projects. Rather than building static structures designed to solve specific problems, coral-inspired approaches might focus on creating adaptive systems that continuously evolve their solutions as problems themselves change. The current crisis facing coral reefs represents both an ending and a beginning. The construction mastery that corals, developed over 500 million years of evolutionary refinement, may not survive the rapid environmental changes of the next few decades. But the principles underlying that mastery, adaptive materials, distributed coordination, emergent intelligence, and multi-scale optimization, offer pathways toward construction capabilities that could transform human civilization. As we stand at the threshold of an era where human activities reshape planetary systems, coral engineering provides a template for creating technologies that work with natural processes rather than against them. The challenge lies not just in understanding how corals build their remarkable structures, but in learning to think like coral colonies themselves, patient, adaptive, persistent, and perpetually optimizing solutions across scales from molecules to ecosystems. The tiny polyps that construct Earth's largest living structures remind us that the most sophisticated engineering often emerges not from grand designs, but from simple rules applied consistently across vast scales of space and time. In their patient, relentless construction activities, corals demonstrate that intelligence itself might be less about individual brilliance than about the wisdom that emerges when countless small decisions align toward common purposes. The conversation between life and matter continues, written now not just in calcium carbonate skeletons growing slowly in tropical seas, but in the growing understanding that the boundary between the living and the built, the intelligent and the mechanical, may be far more fluid than we ever imagined. There is a rule that applies to every living thing that has ever existed on this planet. A rule older than bones, older than eyes, older than the first creature that learned to move. Every organism that has ever drawn energy from its environment, processed it, and used it to build itself has moved in one direction, forward, from simpler to more complex, from young to old, from formation to dissolution. Life assembles, and then it comes apart. This is not a design flaw. Biologists call it senescence, the accumulated damage of living, written into the machinery of cells over time. It is so universal, so embedded into the architecture of life on Earth, that most evolutionary biologists consider it a near certainty. Death is not incidental to biology. It is, in many ways, its engine. Organisms that age and die make room for offspring that can adapt. This is how evolution works. You live, you reproduce, you exit. The species moves forward, even if the individual does not. Almost every organism that has ever existed has followed this rule without exception. Plants, fungi, insects, vertebrates, bacteria colonies, ancient trees with thousands of rings carved into their trunks, all of them bound to the same one-way current of time, biological clocks ticking forward, never back. But in the Mediterranean Sea, drifting among the plankton, there exists a creature so small it disappears against the light, about the size of the nail on your smallest finger. It has no brain, no heart, no lungs. It has existed in roughly its current form for approximately 500 million years. And at some point, in the distant past, this animal worked out something that every other animal on Earth, every mammal, every bird, every plant, every fish, has apparently failed to discover. It worked out how to reverse the direction of its own life cycle, how to take a dying body, unmake its aging cells, and begin again, genetically intact, physiologically young, with no death required. The scientific name of this animal is Turritopsis dohrnii. Its common name tells you everything about why it matters. It is called the immortal jellyfish. This is Omni, stories about life and the mysteries in between. Be sure to like this video and subscribe for more. It is worth pausing on exactly how this was found, because the discovery of biological immortality in a living organism is not the kind of thing a scientist plans for. It is the kind of thing that happens by accident, while someone is doing something else entirely. The summer of 1988. A young German marine biology student named Christian Sommer was spending part of his break on the Italian Riviera in a small coastal town called Rapallo, near the cliffs of Portofino. Sommer was not there purely on holiday. He was working, diving with fine plankton nets in the turquoise water, collecting specimens of a group of animals called hydrozoans. These are ancient, radially symmetrical invertebrates that share a phylum, Cnidaria, with sea anemones and corals. Sommer was interested in their reproductive mechanics, particularly the transition from the sessile polyp phase to the free-swimming adult medusa phase. Among the organisms he netted was a tiny, almost transparent jellyfish with a bright red stomach measuring only a few millimeters across. He brought samples back to his laboratory and placed them in a jar, expecting to observe them mature, spawn, and die. The standard arc of a hydrozoan life cycle. He returned to the jar some days later, expecting to find either dead animals or newly released larvae. Instead, he found something that made no immediate sense. The adult medusae were gone. In their place were small, stationary polyps attached to the bottom of the jar, the juvenile stage that should, by all biological logic, have preceded the adults, not followed them. The animals had not died, they had gone backwards. Sommer reported this finding, and it was initially met with the kind of cautious skepticism that unusual observations tend to attract in science. It was described in a preliminary paper in 1992 by Sommer, Giorgio Bavistrello, and their colleagues. but the scientific community did not immediately grasp the scale of what they had seen. The formal investigation took years. In 1996, a group led by Stefano Piraino at the University of Salento, along with Barrow, Eschbach and Schmid, published a study in the journal Biological Bulletin that documented the process in detail for the first time. Their paper, titled Reversing the Life Cycle, confirmed what Sommer had glimpsed. This jellyfish could consistently, repeatedly, and deliberately return to an earlier developmental stage, not as an accident, not as an aberration, but as a strategy, a response, a choice the body makes when conditions become unfavourable enough. The organism does this in response to starvation, to physical injury, to sudden changes in salinity or temperature, to the simple fact of growing old. When these stresses arrive, the medusa, the adult free-swimming form, does not attempt to fight them. It does not flee. It contracts. Its tentacles are reabsorbed into its body. It loses the ability to swim. It sinks to the seafloor and collapses into a gelatinous, featureless cyst, a blob of undifferentiated tissue with no obvious structure, no apparent function, clinging to whatever surface it can find. This cyst stage lasts between 24 and 36 hours, and within that window, something happens that has no real parallel in complex animal biology. The cells inside that cyst rearrange themselves. They communicate. They change. And what emerges from the cyst is a new polyp, a juvenile jellyfish, ready to grow again, capable of budding off an entire new colony of genetically identical medusae. The animal does not die. It resets. To appreciate why this matters, it helps to understand what a cell normally does and what it is normally capable of. When a complex organism like a jellyfish, or a human being, or a frog, or a tree develops from a fertilized egg, it begins as a single cell with a complete genetic blueprint. As it divides, its daughter cells begin to specialize. Muscle cells switch on the genes that make them contractile and switch off everything else. Nerve cells activate the machinery of electrical signaling and suppress the pathways for, say, digestion. Skin cells build keratin and form barriers. Each cell, as it matures, becomes more committed to its function and less capable of becoming something else. This process, differentiation, is normally considered to be a one-way road. A neuron does not decide one morning to become a liver cell. A muscle fibre does not transform into an eye cell. The commitment is, in nearly all complex animals, permanent. Transdifferentiation violates this principle. The word itself means approximately what it sounds like. Differentiated cells transforming into different differentiated cells without passing through an undifferentiated intermediate stage. In Turritopsis dohrnii, the specialized cells of a dying adult medusa, cells built for swimming, for digestion, for reproduction, appear to shed those commitments, revert toward a more primitive state, and then respecialize into the very different cell types needed to build a polyp. A mature cell, in effect, forgets what it is and becomes something else. This process, as Dr. Maria Miglietta, Associate Professor of Marine Biology at Texas A&M University at Galveston, and one of the world's leading researchers on the species, has noted, is capable of occurring in less than 24 hours. The entirety of the animal's adult identity can be dismantled and rebuilt in roughly one day. It accomplishes this without a brain, without a nervous system of any sophistication, without lungs, without any of the dedicated organs that most animals rely on to regulate their biology. The system appears to be cellular, distributed, emergent, driven not by a central command but by molecular signals passing between individual cells. This has no precise equivalent in any other animal we know of. There are organisms that regenerate lost limbs, salamanders, planarians, certain starfish. There are organisms that can reproduce from fragments of their own bodies. There are organisms that can enter states of near-suspended animation and revive years later. But these are partial examples. They regenerate specific tissues. They survive dormancy. They do not accomplish what Turritopsis dohrnii accomplishes, which is the complete reversal of an adult organism's developmental trajectory, the wholesale reprogramming of its own cellular identity, not once, but indefinitely, as a repeatable survival strategy. Shin Kubota, a scientist who spent decades at Kyoto University's Sito Marine Biological Laboratory before founding the Turritopsis, Immortal, Jellyfish Regenerative Biological Research Institute in Arita Wakayama, kept colonies of Turritopsis dohrnii alive in controlled conditions for years. Across a 2-year observation period documented in his published work, he observed individual colonies of these jellyfish cycling through the reversal process up to 11 times, each time regenerating successfully, each time producing new medusae. The only interruptions were practical ones: the difficulty of keeping the animals alive in artificial conditions, the need to feed them minuscule portions of brine shrimp prepared under a microscope, the sensitivity of the animals to even minor fluctuations in water quality. Given optimal conditions, there was no observed biological limit to how many times the cycle could repeat. There is something worth dwelling on in the physical reality of what the cyst stage actually looks like, because it defies easy intuition. When Turritopsis dohrnii initiates its reversal, the transformation is not gradual. The animal does not slowly age backward, it collapses. The bell, the umbrella-shaped body of the adult Medusus, contracts inward. The ring of tentacles that it uses to capture prey is retracted and absorbed into the body wall. The animal loses all locomotive function, it sinks, and what settles on the seafloor, or on whatever hard substrate is available, is something that looks to the naked eye like nothing in particular: a small, structureless mass, a biological smear. Nothing about its appearance suggests that it contains the instructions for an entire jellyfish. Inside that cyst, what is actually occurring is one of the most radical molecular reorganizations observed in any animal. The specialized tissues of the medusa, the epithelial cells of the body wall, the contractile muscle cells around the bell margin, the cells of the gastrovascular cavity used for digestion, the germline cells responsible for reproduction, all of these populations are changing. They are not dying in the way that cells normally die. They are not undergoing apoptosis, the controlled cell death that multicellular organisms use to remove damaged or unnecessary cells. Instead, they appear to be shedding their functional commitments while remaining alive. The proteins that define what type of cell, each one is the molecular markers of identity, the transcription factors that keep certain genes switched on and others switched off, begin to shift. Cell types that should be fixed in their function begin acquiring characteristics of other cell types, or of more primitive cell types without a specific function at all. The result is a transient state of cellular ambiguity. A muscle cell from the bell of the medusa may no longer be entirely a muscle cell, but it is not yet whatever it is becoming. This is the window during which transdifferentiation occurs, not the neat instantaneous conversion of one cell type into another, but a messy overlapping transition through intermediate states that have no precise parallel in normal animal development. What makes this possible without catastrophic error is the suite of molecular chaperones, DNA repair complexes, and epigenetic regulators that the genome of Turritopsis dohrnii maintains in expanded, enhanced forms compared to related species. The machinery of reprogramming requires protection. Without it, the genetic blueprint the cells are reverting to would be too degraded to produce a functional organism on the other side of the transformation. When the cyst begins to resolve into a polyp, the process runs in reverse. Cells commit to the roles required by the juvenile stage. The stolon, the root-like base of the polyp, forms first, anchoring the animal. The body column extends. The first tentacles emerge, smaller and differently configured than the adult medusa tentacles they replaced. The feeding apparatus forms, and within days the animal is a functional juvenile once again, capable of feeding, growing, and eventually budding off new medusae into the water column above it. Each of those medusae carries the original genome intact. They are, genetically, the same organism that began the reversal. The information has been preserved across a transformation that dissolved and reconstructed the entire physical body. For several decades, the description of this phenomenon remained largely observational. Scientists could document what the jellyfish did. What they could not fully explain was how. The molecular machinery behind transdifferentiation in Turritopsis dohrnii was not mapped in detail until more recently, and when it finally was, the findings complicated the story in important ways. In August of 2022, a Spanish research team published the results of the most comprehensive genomic study of Turritopsis dohrnii conducted to that point. Their work appeared in the Proceedings of the National Academy of Sciences, one of the most widely read scientific journals in the world. The team, which included Maria Pascal-Torner and colleagues, did something methodologically elegant. They sequenced and compared two whole genomes side by side. The first was Turritopsis dohrnii, the immortal species. The second was Turritopsis rubra, a closely related jellyfish that shares much of the same ancestry, lives in similar environments, but does not rejuvenate. It ages and dies. By comparing these two genomes across approximately 1,000 genes associated with aging, DNA repair and cellular maintenance, the team was able to identify specific molecular differences that appear to confer immortality on one species and deny it to the other. The differences were not simple. There was no single gene, no master switch, no isolated mutation that explained everything. Instead, the Spanish team found a suite of variations, many genes, many pathways, all working in concert. Turritopsis dohrnii carried significantly expanded copies of genes involved in DNA repair and DNA replication, suggesting the species has a substantially enhanced capacity to maintain the integrity of its genetic material across cycles of cell division and reprogramming. It carried more copies of genes involved in managing oxidative stress, the reactive oxygen species that accumulate inside cells over time and damage proteins, lipids, and genetic material. The fact that the immortal jellyfish appears to neutralize these reactive compounds more effectively than its mortal relative may explain in part why its cells can tolerate the radical transformation of transdifferentiation without catastrophic genetic damage. Critically, the Spanish team also identified changes in telomere maintenance. Telomeres are the protective caps at the ends of chromosomes, sequences of repetitive DNA that shorten slightly every time a cell divides. In most organisms, telomere shortening is one of the primary mechanisms of cellular aging. As telomeres erode past a threshold, cells enter a state called senescence, in which they stop dividing, begin releasing inflammatory signals, and eventually die. Turritopsis dohrnii appears to carry variants of telomere-associated genes that may allow it to better maintain or restore these protective caps during the reversal process. Then there is what happened to gene expression during the reversal itself. The research team found that when Turritopsis dohrnii initiates life cycle reversal, it activates targets associated with pluripotency, the capacity to become any type of cell, while simultaneously silencing a molecular complex called polycomb repressive complex 2. This complex normally acts as an epigenetic lock, keeping cells committed to their differentiated state by suppressing the genes associated with embryonic development. When it is silenced, those ancient developmental programs can switch back on. The cell can hear instructions it had long stopped listening to. Earlier transcriptome research from Texas A&M University, led by Dr. Milieta and published in G3, Genes, Genomes, Genetics in 2019, had found that during the cis stage, that featureless blob between medusa and polyp, genes related to aging and lifespan regulation were significantly enriched, while genes associated with mitotic cell division were suppressed. The animal was not proliferating wildly, it was repairing, maintaining, resetting. It was prioritizing the quality of its DNA over the quantity of its cells. This is a key distinction. Transdifferentiation in Turritopsis dohrnii is not runaway cellular growth. It is controlled cellular re-education. Subsequent research from the Kazusa DNA Research Institute in Japan, published in 2022 with contributions from Shin Kubota himself, assembled a full genome sequence of Turritopsis dohrnii from 1,500 medusae originating from a single clone. The team identified 23,314 high-confidence genes and analysed their expression patterns across developmental stages, further confirming the unusual activation of pluripotency-associated genetic networks during the reversal process. The molecular portrait that emerges from all of this is not of a single miraculous adaptation, but of a deeply integrated biological architecture, one that has been refined, expanded, and reinforced across millions of years of evolution to maintain genomic stability through a process of cellular reprogramming that would simply destroy most complex animals. And so, a question becomes unavoidable. If this works, if it genuinely works, as the evidence suggests it does, then why is Turritopsis dohrnii essentially alone in doing it? Why hasn't natural selection spread this adaptation more widely? Why, across 500 million years, has biological immortality through transdifferentiation remained the property of one tiny brainless jellyfish and almost nothing else? This question cuts to the centre of how evolution actually operates, and the answer reveals something important about the nature of biological systems and the trade-offs that govern them. The first thing to understand is that Turritopsis dohrnii is not, in any absolute sense, immune to death. It can be eaten, and frequently is. It can be killed by environmental changes severe enough to exceed even its considerable resilience. What it has solved is specifically death by aging, death by the internal accumulation of damage, predation, infection, habitat collapse. These remain existential threats. Biological immortality, in this context, is a conditional concept. It means the organism has removed one of the many possible causes of death, not all of them. The second thing to understand is that immortality is not obviously advantageous from an evolutionary perspective. This sounds counterintuitive. Surely living forever is better than dying. But natural selection does not favour what is good for an individual. It favours what is good for genetic propagation in a given environment. Organisms that produce many offspring quickly and then exit can, in the right conditions, outcompete organisms that live a long time but reproduce slowly. Aging paradoxically may have been selected for because it clears genetic material from the population, fast enough for adaptation to keep pace with changing environments. Immortal, organisms would accumulate older genetic configurations in the gene pool, potentially making populations less responsive to environmental pressure. Turritopsis. Dorney's solution may represent a narrow evolutionary path that works specifically for a small, simple organism with minimal energy demands, living in an environment where physical destruction by predation is the more immediate threat than any other cause of death. For larger, more complex organisms, especially those with sophisticated nervous systems, immune systems, and metabolic machinery, the costs of maintaining the cellular stability required for transdifferentiation may simply outweigh the benefits. The infrastructure needed to reprogram cells safely, to maintain genomic integrity through repeated cycles of reversal, appears to be substantial. It requires an expanded set of DNA repair enzymes, enhanced antioxidant systems, sophisticated epigenetic regulation, and a telomere maintenance system that most animals simply do not possess. There's a deeper problem as well. Transdifferentiation, the reprogramming of differentiated cells, is a process that, in most complex animals, including humans, is associated not with rejuvenation, but with disease. When human cells undergo something resembling transdifferentiation, the usual outcome is cancer. The polycomb repressive complex that Turritopsis dornii suppresses during reversal is the same type of complex whose disruption is associated with tumour formation in human biology. The activation of pluripotency genes that gives the jellyfish the ability to rebuild itself is, in the wrong cellular context, precisely the kind of genetic event that allows cancer cells to proliferate without limit. Research published in late 2024 in the Revista Española de Geriatría y Gerontología by researchers including collaborators from the Weill Cornell Medical College and the Pontificia Universidad Javeriana in Bogotá explored exactly this contrast. In Turritopsis dornii, DNA, synthesis and related repair genes are overexpressed during cellular reprogramming, while genes related to cell division are suppressed. DNA quality takes priority over cellular replication. In humans, when damaged DNA accumulates, particularly when telomeres shorten and reactive oxygen species increase, the conditions that trigger something resembling transdifferentiation are the same conditions that promote oncogenic activity and metastatic spread. The jellyfish has solved a problem that, for us, creates a different and equally serious problem. The distinction appears to lie in the integrity of the genome at the moment of reprogramming. Turritopsis dornii initiates its reversal before its DNA is significantly compromised. It responds to stress proactively, not reactively. Its enhanced DNN repair machinery ensures that when pluripotency genes are activated, the genetic blueprint the cells are reverting to is still coherent, still accurate, still capable of producing a functional organism. In human aging, by contrast, the accumulation of DNA damage precedes any attempt at cellular reprogramming, meaning the blueprint available to reprogram cells is already corrupted. This is why there is no single gene for immortality in Turritopsis dornii. There is no secret ingredient. There is a whole system, a coordinated network of cellular maintenance strategies that took hundreds of millions of years to assemble, that together create the conditions under which transdifferentiation can occur safely and successfully. To understand what makes Turritopsis dornii genuinely singular, it helps to look at the other organisms that have attracted attention from aging researchers, the other candidates, the partial solutions, the near misses of biological immortality. because nature has produced several extraordinary experiments in longevity. None of them accomplish what this jellyfish accomplishes, but examining why they fall short sharpens the picture of what transdifferentiation actually represents. Begin with the Hydra, a genus of small freshwater polyps, animals distantly related to Turritopsis dohrnii within the phylum Cnidaria. Hydra polyps are structurally simple, a tube-shaped body with a ring of tentacles at one end, but what makes them biologically extraordinary is that their cells turn over almost continuously, driven by a population of stem cells concentrated in the middle of the body column. These stem cells divide constantly, producing fresh differentiated cells that migrate outward and replace older ones. The organism is, in effect, perpetually renewing itself from a core of permanent juvenile cells. Research by Daniel Martinez at Pomona College, published in the late 1990s and replicated in subsequent studies, found no detectable increase in mortality rate in Hydra over observation periods of several years. No statistical evidence of aging. In a 2012 study published in the Proceedings of the National Academy of Sciences, Martinez and colleagues confirmed that Hydra do not appear to age in the conventional sense, and that their sustained stem cell proliferation is maintained by elevated expression of a transcription factor called FOXO, a gene involved in stress resistance and longevity that is also found in slightly different form in a wide range of organisms, including humans. But Hydra do not reverse their development. They do not reprogram their cells. They maintain their youth through continuous replacement rather than through transformation. A Hydra that is injured, starved, or environmentally stressed does not collapse into a cyst and reconstitute. It draws on its stem cell reservoir and rebuilds lost tissue locally. The mechanism is impressive, but it's more analogous to deep wound healing than to Turritopsis dohrnii's whole body reversal. Hydra also remain structurally simple. They do not develop into radically different morphological stages. There is no Medusa. There is no complex life cycle to reverse. What they have is a maintenance strategy. What Turritopsis dohrnii has is a reset strategy. Then there are the lobsters. American lobsters, Homarus americanus, have attracted considerable popular attention for their apparent resistance to the usual signs of aging. Unlike most animals, mature lobsters show no measurable decline in reproductive capacity, strength, or metabolic function with age. They continue to grow throughout their lives. They continue to molt, shedding their shells and producing new ones. The mechanism behind this is partly related to an enzyme called telomerase, which rebuilds the telomeric caps at the ends of chromosomes that shorten with each cell division. Lobsters express telomerase in most of their tissues throughout their lives, not just in the stem and germline cells where most animals confine it. This allows their cells to maintain telomere length and continue dividing without the progressive. Shortening that leads to senescence in most organisms. Lobsters, however, do eventually die, and not of old age in any simple sense. They die because molting becomes metabolically unsustainable at very large body sizes. The energy cost of producing a new shell increases with every molt, and at some point, the animal's metabolic systems cannot keep pace. They also die from accumulated disease, infection, and the physical damage of a marine environment. Their telomerase expression buys them something significant, a resistance to replicative senescence, but it does not confer the ability to reverse or reset their developmental trajectory. They age slowly and impressively, but they age. The naked mole rat occupies a similar position in the longevity literature. These small burrowing rodents live 10 times longer than other mammals of comparable size, show remarkably low cancer rates, and maintain their reproductive capacity and physical function into very late life. Research into their biology has identified several contributing factors. Unusually high-fidelity protein synthesis that produces fewer misfolded proteins than other rodents, an enhanced dsNA repair system, high concentrations of a particular form of the sugar hyaluronic that may suppress tumor development, and a distinctive cellular response to stress. But naked mole rats age. They accumulate damage. Their tissues deteriorate. They simply do it far more slowly than their phylogenetic relatives, and the mechanisms by which they resist aging are about prevention and maintenance rather than reversal. What these organisms share is that they are resisting the damage of living, suppressing it, slowing it, working around its worst consequences. What Turritopsis dohrnii does is categorically different. It does not resist cellular aging. It undoes it. The distinction sounds semantic. It is not. An organism that resists aging must maintain a continuous battle against entropy across its entire lifespan, expending energy to repair damage at roughly the rate damage accumulates. An organism that can reverse aging needs only to reach a threshold at which reversal becomes necessary, then execute a wholesale transformation that begins the process again from a refreshed biological baseline. The two strategies make different demands on the organism, produce different vulnerabilities, and represent different evolutionary solutions to the same underlying problem. One slows the clock, the other resets it. There is a postscript to the story of Turritopsis dohrni's biology that says something remarkable about what happens when evolution invests this heavily in an organism's survival. The animal was first formally catalogued in Naples in 1883. For most of the following century it was considered a Mediterranean species, small, obscure, ecologically unremarkable. By the time Christian Sommer netted his specimen in 1988, the animal was known only from European and Japanese waters, and even then, only to specialists in hydrozoan taxonomy. Today, Turritopsis dohrni is found in every temperate and tropical ocean on Earth. It has been documented off the coasts of Spain, Japan, the Mediterranean basin, the southeast United States and Panama. It appears in the Pacific. It appears in the Atlantic. Populations have been recorded in waters where it had never historically been observed, and the genetic evidence suggests that in most of these locations, it arrived not through natural migration but aboard ships, surviving in ballast water tanks, suspended in that intermediate cis state that allows it to endure conditions that would kill most marine invertebrates. This spread, documented in detail by Dr. Maria Milieta during years of global fieldwork beginning during her graduate studies at Duke University, has been described as a silent invasion. Silent because, unlike most invasive species, Turritopsis dohrni does not appear to devastate the ecosystems it enters. It has no obvious large-scale predatory impact. It drifts, it persists, it cycles through its reversals, largely unnoticed. What makes this migration significant is less the ecological consequence than the biological demonstration it provides. This animal, precisely because it can survive conditions that would kill ordinary jellyfish, starvation, physical trauma, hostile water chemistry, can travel to places that its life cycle would otherwise make impossible. Its immortality is, in a practical sense, also its passport. The same mechanism that keeps individual animals alive is the mechanism that allowed the species to colonize the world's oceans over the course of a century or two of maritime shipping. When a single Turritopsis dohrni polyp reaches a new coastline, it can bud off multiple new medusae, each genetically identical to the original. Those medusae can themselves reverse and rebud. From one ancestor that survived a journey no ordinary jellyfish would survive, an entire local population can emerge. Dr. Milieta's research has pointed out that a single immortal polyp colony can produce hundreds of new individual jellyfish, injecting genetic copies of itself into whatever environment it reaches. The mathematics of this are quietly extraordinary. The most obvious question any thoughtful person brings to this story is also the most complicated one. Does any of this translate to human medicine? Is there a path from Turritopsis dohrni to something practically useful for human aging? The honest answer from the scientists who study it most carefully is not directly and not soon. But the implications of the research are not trivial. The concept at the centre of modern regenerative medicine, the idea that differentiated adult cells can be reprogrammed to become something else, is the same concept that Turritopsis dohrni executes naturally, reliably, and without apparent limit. The Yamanaka factors, a set of 4 genes identified by Shinya Yamanaka at Kyoto University, are capable of reprogramming adult mammalian cells back to a pluripotent state. The discovery that earned Yamanaka the Nobel Prize in Physiology or Medicine in 2012. Research into the transcriptome of Turritopsis dohrni, the full set of genes being actively expressed at each life stage, has found evidence of similar pluripotency-associated factors at work during life cycle reversal, including gene families analogous to the Yamanaka factors and related Thompson factors. The jellyfish appears to have evolved its own version of a reprogramming toolkit and learned, over geological time, how to use it without triggering the genomic instability that makes cellular reprogramming dangerous in mammalian systems. Understanding how it manages this, how it maintains DNA integrity during reprogramming, how it silences and reactivates epigenetic programs with precision, how it coordinates telomere maintenance across a whole body transformation, represents a legitimate scientific frontier. Researchers at institutions including Texas A&M, Cornell, universities in Spain, Japan and Colombia are actively pursuing these questions. The research published in PNAS in 2022 laid a genomic foundation that makes the next round of molecular investigation significantly more tractable. What's already clear is that the jellyfish's approach to cellular aging offers a conceptual alternative to the frameworks that dominate human aging research. Most anti-aging research focuses on slowing damage accumulation, using antioxidants, senolytics, drugs that clear senescent cells, telomere-extending therapies, and caloric restriction pathways to extend healthy lifespan by reducing the rate at which cells deteriorate. Turritopsis dohrnii takes a different approach entirely. Rather than slowing the damage clock, it resets it. Rather than maintaining an aging system, it discards the aging system and builds a new one from the same genetic blueprint. This does not mean the jellyfish's strategy is translatable to humans in any straightforward sense. The organizational complexity of a human body, trillions of cells, specialized across hundreds of distinct tissue types, organized into elaborate hierarchies of organs and organ systems, governed by an immune system that actively monitors and responds to cellular identity, makes anything like whole-body transdifferentiation not just practically impossible, but conceptually incoherent. You cannot reprogram a human nervous system in the way you reprogram a jellyfish with 800 cells. The jellyfish has no neurons that store decades of learned information. It has no immune memory that would be lost and disorienting if reprogrammed away. It has no continuous subjective experience that would raise difficult questions about identity across resets. But targeted cellular reprogramming, selectively applying the principles of transdifferentiation to specific tissues, specific organs, specific cell populations that have accumulated damaging senescence, is an active area of investigation. And the jellyfish's genomic toolkit for doing this safely, its expanded suite of DNA repair enzymes, its telomere maintenance variants, its ability to suppress polycomb complexes in a controlled rather than catastrophic way, represents a library of biological solutions that evolution has already tested across hundreds of millions of years. The parallel between what Turritopsis dohrnii does and what Yamanaka factor research is attempting to do is not accidental, and researchers are increasingly explicit about it. Partial reprogramming, the application of Yamanaka factors to mammalian tissues for brief, controlled intervals, rather than the full duration needed to produce a pluripotent stem cell, is one of the most actively investigated strategies in aging biology today. The logic is that a short pulse of reprogramming signals might reset epigenetic aging markers in a cell, without erasing its functional identity, producing something like cellular rejuvenation, without the loss of differentiated function. Companies including Altos Labs, founded in 2021 with substantial investment from some of the world's wealthiest research backers, are pursuing exactly this approach in mammalian systems. What they are attempting to do artificially, in the controlled environment of a laboratory, Turritopsis dohrnii does naturally, repeatedly, and across its entire body. The jellyfish is not merely a curiosity adjacent to this research. It is, in a meaningful sense, its most compelling proof of concept. Evidence that cellular reprogramming at scale, executed safely, is biologically achievable. The question that drives the field is not whether it can happen. The jellyfish answers that. The question is whether the specific molecular architecture that makes it safe in this animal can be reverse engineered, adapted and applied in organisms of vastly greater complexity. Dr. Milieta has put this plainly in her research communications. Understanding how cells can reprogram themselves to become something else that is needed is foundational to regenerative medicine. And Turritopsis dohrnii is, as she has noted, the only creature known to accomplish full life cycle reversal in less than 24 hours. That speed, that efficiency, that completeness is itself a data point. Evidence that cellular reprogramming does not have to be a slow, imprecise, or inevitably dangerous process. It can, under the right conditions, be rapid, controlled, and survivable. There is a question that lies beneath the biology, one that no genome study can fully resolve. It is the kind of question that the sciences tend to generate at their edges, where observation shades into something larger. the conventional view of aging in biology is that it is not a flaw, it is a feature. Senescence, from this perspective, is evolution's way of clearing the field, removing older, genetic configurations to make room for new ones that may be better adapted to a changing world. Organisms that age and die contribute their bodies to the nutrient cycle. Their death feeds other life. Their passing is not incidental to the ecosystem, it is part of its function. Teratopsis dorni does not play by these rules. It does not clear the field. It stays. It cycles. It produces copies of itself from a blueprint that does not substantially change across cycles of reversal. From one perspective, this looks like a refusal, a quiet biological protest against the entropy that governs everything else alive. From another perspective, it looks like what evolution always looks like when you examine it closely, an improvisation that happens to work in a particular environment, under a particular set of pressures, without any inherent moral weight attached to it. But it does raise a genuine philosophical puzzle about the nature of aging itself. If aging is selected for, if it persists because it benefits genetic propagation, then why does Teratopsis dorni seem to benefit genetically from avoiding it? The answer, most likely, is that the benefit/cost calculation is genuinely different for a small marine invertebrate in an ocean full of predators than it is for a large terrestrial vertebrate in a social environment. The jellyfish's solution is not a universal solution to aging. It is a solution to the specific form of mortality that was most dangerous for Teratopsis dorni in the environment it evolved in. But it demonstrates that the one-way directionality of biological time is not an absolute law. It is a tendency, a deeply reinforced near-universal tendency, but not a law. There is also the question of what it means to be the same organism across a cycle of complete cellular reprogramming. When a Teratopsis dorni medusa collapses into a cyst and reconstitutes as a new polyp, the genetic material is continuous. The DNA in the polyp is the same DNA that was in the medusa, but the cells are different, the body plan is different, the stage of development is different. The organism retains its genetic identity across the reversal, but not its cellular identity. Whether it retains anything else, whether there is any meaningful continuity of biological individuality beyond the genome, is a question that the biology alone cannot settle. This is the ship of Theseus problem written in living tissue. If every plank of the ship is replaced, is it still the same ship? If every cell in a jellyfish changes its identity, is it still the same jellyfish? The answer depends entirely on what we decide constitutes identity in a biological system. For a creature with no nervous system of any significant complexity, no learned behaviour, no memory in any meaningful neurological sense, the question may ultimately be unanswerable, and possibly irrelevant. The organism functions, the genome persists, the cycle continues. For an organism like a human, the same question would carry infinitely more weight. The story of Teratopsis dorni is not really a story about immortality, or rather, it is a story about immortality that forces a reconsideration of what immortality means, and why the concept is so much more interesting when stripped of mythology and examined as a biological question. The jellyfish does not live forever. It cannot. The ocean is full of things that will eat it. The ocean is full of conditions it cannot survive. Immortality, in the absolute sense that human beings have spent centuries dreaming about, is not what this animal possesses. What it possesses is something more specific, more technically interesting, and arguably more philosophically provocative. A mechanism for removing one category of death. The death that comes from the inside, from the accumulation of damage, from the slow entropic unwinding of cellular machinery, while remaining fully subject to everything else the world might bring. In this narrower sense, it solved something real, something specific, something that 500 million years of animal evolution have, in every other lineage, apparently decided was not worth solving, or not possible to solve without costs too high to bear. The fact that the solution exists at all is significant. It means that the cellular machinery of aging is not an absolute constraint on life. It is a biological condition that has at least one known exception. And that exception, drifting in ocean currents from the Mediterranean to the Pacific, is 4.5 millimeters wide, has no brain, and has been cycling through its own quiet reversals long before there were eyes in the sea capable of watching it do so. The research that began from Christian Summers' accidental observation in a coastal jar in 1988, that was confirmed by Perino and colleagues in 1996, expanded by Shin Kubota across decades of patient observation, and illuminated at the Genomic level by Spanish researchers in 2022, has not yet produced a therapy for human aging. It has not produced a drug, a protocol, a single clinical application. What it has produced is a window into a form of biology that was not supposed to exist, into a molecular architecture that solves a problem every other complex organism has accepted as unsolvable. Scientists are still working at that window, the full epigenetic program driving transdifferentiation in Turritopsis dohrnii, which specific marks are laid down, which are erased, how the timing is coordinated across the cells of the cyst, remains an active area of research. Institutions including Bristol University's SW Biodoctoral Training Programme have ongoing projects specifically aimed at mapping these epigenetic changes in detail. The PNAS genomic study of 2022 was a foundation, not a conclusion. What is already clear is that this animal exists, that it does what the evidence says it does, that its genome reveals a coherent, integrated biological strategy for cellular maintenance across reversal cycles, that the strategy is complex, expensive to maintain, and apparently achievable only because it was built incrementally across geological time by the same evolutionary pressures that built everything else that lives. Life, on this planet, has been running its experiments for approximately 4 billion years. Most of those experiments end in extinction. Some produce organisms that persist for millions of years in forms that change slowly, and a very small number produce something that appears, from the outside, to violate the rules, that represents a path not taken by the vast majority of living things, a direction so improbable that when it is finally observed, it takes a generation of scientists to fully accept that the observation is real. Turritopsis dohrnii is one of those experiments, an ancient one, quiet, nearly invisible, drifting in every ocean on Earth by now, cycling through, reversals in the silty dark of harbour floors and open water alike. It has been doing this since long before anything with a backbone existed, long before the first fish looked up and saw the surface of the water, long before the first limb reached toward air. It figured something out, and then it kept it to itself. Hidden in the mechanics of a body too small to see without magnification, encoded in a genome that no one thought to read until the 21st century. The secret was always there. It just required someone to stop and look more carefully at a jar full of very small animals that should have died but didn't,